Hey everyone, tonight we're diving into a story that began when Earth was a completely alien world. No green forests, no blue skies, no animals of any kind. Just a hostile, volcanic planet, covered in toxic oceans under a methane-filled atmosphere. And yet, somewhere in this hellscape, something extraordinary happened. Life began. Not the complex life we know today, but the first tiny, simple cells that would change everything forever. So before we journey back 3.5 billion years, take a moment to hit that like button if you're ready for some serious time travel, and let me know in the comments where you're watching from and what you think about when you imagine Earth's earliest days. Now, settle in, maybe grab a warm drink, and let's explore the most important chapter in our planet's history together. Picture Earth 3.5 billion years ago. The sun burns dimmer in the sky, providing about 30% less energy than it does today. The atmosphere is a suffocating mix of methane, carbon dioxide, and hydrogen, with virtually no oxygen to breathe. The oceans are not the familiar blue we know, but a murky chemical soup filled with iron and sulfur compounds that would be toxic to almost every living thing on modern Earth. Volcanic activity dominates the landscape, with underwater vents spewing superheated, mineral-rich water into the primitive seas. This is the Archean Eon, a time when our planet looked more like an alien world than the vibrant biosphere we call home. But in this seemingly impossible environment, chemistry was working its magic. Deep beneath the ocean's surface, where crushing pressure and intense heat created conditions unlike anywhere else on Earth, the building blocks of life were assembling themselves into something remarkable. The story we're about to tell isn't science fiction. It's the documented, evidence-based account of how life emerged from the chaos of early Earth, preserved in rocks that have survived billions of years of geological upheaval. The evidence for Earth's earliest life comes from some of the most remote and rugged places on our planet. In the red, dusty landscape of Western Australia's Pilbara Craton, geologists have uncovered what may be the most precious rocks on Earth. These ancient formations, dating back 3.5 billion years, contain the fossilized remains of Earth's first living communities. The preservation is nothing short of miraculous, through a combination of rapid mineralization and protection from subsequent geological destruction. These rocks have maintained their secrets for longer than most people can truly comprehend. What scientists found in these ancient rocks was revolutionary. Layer upon layer of stromatolites, distinctive laminated structures that form when microbial communities trap sediment and precipitate minerals as they grow. These aren't random mineral formations. They're the architectural legacy of organized biological communities, complex ecosystems of microorganisms working together in ways that mirror some of the processes we still see in living microbial mats today. The discovery didn't happen overnight. Researchers spent decades carefully analyzing these formations, using advanced chemical techniques to distinguish between purely geological processes and biological signatures. They examined isotope ratios, looking for the telltale signs of life's preference for lighter carbon atoms. They studied microscopic structures, searching for cellular remains preserved in the mineral matrix. They compared ancient formations with modern analogues, like the living stromatolites still growing in Australia's Shark Bay. Every test pointed to the same conclusion. These weren't just rocks. They were fossils of Earth's first ecosystems, communities of single-celled organisms that had learned to harness energy from their environment and organize themselves into structured, cooperative societies. The implications were staggering. Life hadn't just appeared on early Earth. It had thrived, building complex communities and leaving behind a geological record that would survive for billions of years. The organisms responsible for these ancient structures were prokaryotes, single-celled life forms fundamentally different from the complex cells that make up plants, animals, and fungi today. Unlike modern eukaryotic cells, these early organisms lacked membrane-bound nuclei, specialized organelles, and the intricate internal machinery that characterizes more advanced life forms. But what they lacked in complexity, they made up for in metabolic versatility and resilience. These early prokaryotes were chemical innovators, developing metabolic pathways that could extract energy from the harsh conditions of Archean Earth. Some learned to process sulfur compounds spewing from hydrothermal vents. Others developed primitive forms of photosynthesis, using sunlight to drive chemical reactions that would eventually transform Earth's atmosphere. They reproduced through simple cell division, creating exact copies of themselves without the genetic recombination that characterizes sexual reproduction in more complex organisms. Despite their apparent simplicity, these prokaryotic communities displayed remarkable sophistication in their organization. They formed microbial mats, multi-layered sheets of different species arranged according to their specific metabolic needs and environmental tolerances. The upper layers might contain organisms that could tolerate or even require oxygen, while deeper layers housed species that thrived in oxygen-free conditions. Each layer had its role in the community's overall metabolism, creating a biological system that was greater than the sum of its parts. The preservation of these ancient microbial communities required extraordinary circumstances. As organisms died and were buried by sediment, rapid mineralization had to occur before decay could destroy cellular structures. The chemical conditions had to be just right to allow minerals to replace organic material while maintaining the original shapes and arrangements. Then, for billions of years, these fossil deposits had to survive the geological violence of a dynamic planet, avoiding the heat and pressure that would have erased their delicate structures. The fact that we can still study these fossils today speaks to both the robustness of early microbial life and the remarkable stability of certain geological formations. The Pilbara Craton represents one of Earth's most ancient and stable crustal blocks, a geological time capsule that has remained relatively unchanged while most of the planet's surface has been recycled through tectonic processes. This stability made it possible for 3.5 billion-year-old biological signatures to survive into the modern era. But the Pilbara isn't the only window into early life. Similar discoveries have been made in South Africa's Barberton Greenstone Belt, where 3.4 billion-year-old microfossils provide additional evidence for ancient prokaryotic communities. These sites, scattered across different continents, suggest that early life wasn't confined to a single location, but had achieved a global distribution by the Middle Archean period. The chemical signatures preserved in these ancient rocks tell a story that goes beyond simple presence or absence of life. Isotope analysis reveals details about early metabolic processes, showing how these organisms processed carbon and other elements. The ratios of different carbon isotopes in ancient organic matter match the patterns we see in modern biological systems, providing strong evidence that similar metabolic processes were operating billions of years ago. These chemical fingerprints are particularly important because they can survive even when physical structures are destroyed by geological processes. In rocks where no visible fossils remain, isotopic signatures can still provide evidence for ancient biological activity. This has allowed scientists to push back the timeline of life's emergence even further, with some chemical evidence suggesting biological processes may have been operating as early as 4.1 billion years ago. The organisms living 3.5 billion years ago faced challenges that would be unimaginable to modern life forms. The atmosphere contained virtually no free oxygen, forcing all metabolic processes to operate under anaerobic conditions. Carbon dioxide levels were hundreds of times higher than today, creating a powerful greenhouse effect that was necessary to keep the planet's surface warm enough for liquid water under the dimmer young sun. The oceans themselves were chemically alien, rich in dissolved iron and sulfur compounds that would be toxic to most modern organisms. Yet these harsh conditions weren't obstacles to early life, they were opportunities. The chemical energy available in these environments provided fuel for metabolic processes that could operate without sunlight or organic matter from other organisms. Early life learned to eat the planet itself, extracting energy from the chemical gradients and mineral reactions that characterize the young Earth. This metabolic flexibility was crucial for life's early success and expansion. Unlike modern ecosystems that depend heavily on photosynthesis and the complex food webs it supports, early microbial communities could thrive in environments where sunlight never penetrated deep ocean vents, underground water systems, and the spaces between mineral grains all became potential habitats for organisms that could derive energy from chemical reactions. The development of these diverse metabolic pathways represents one of life's greatest early innovations. Different species of prokaryotes evolved to specialize in processing different types of chemical energy, creating the potential for complex ecological relationships even in these early communities. Some organisms might produce waste products that served as food for others, establishing primitive food webs based entirely on chemical cycling rather than solar energy. These early metabolic innovations laid the foundation for all subsequent life on Earth. The biochemical pathways that evolved in Archean prokaryotes remain essential components of modern cellular metabolism. Even today, many of the most fundamental processes of life, from DNA replication to protein synthesis, operate according to principles that were first established in these ancient microbial communities. The story of life's emergence isn't just about biological evolution, it's about the co-evolution of life and planet. As early organisms grew and reproduced, they began to alter their environment in ways that would have profound consequences for the future of Earth. Their metabolic activities changed the chemistry of the oceans and atmosphere, setting in motion processes that would eventually make the planet more hospitable to complex life forms. But we're getting ahead of ourselves. Before life could transform the planet, it first had to solve the fundamental challenge of coming into existence in the first place. The transition from complex chemistry to living, reproducing organisms represents one of the most important and mysterious chapters in natural history. And the evidence suggests this transition happened not in shallow tide pools or lightning-struck ponds, but in the extreme environments of underwater volcanic vents, deep beneath the churning surface of ancient oceans, where crushing pressure and searing heat created conditions that would instantly destroy most modern life. Nature was conducting its most ambitious experiment. Hydrothermal vents, those towering underwater chimneys spewing mineral-rich water at temperatures reaching 350 degrees Celsius, weren't just geological curiosities. They were chemical laboratories where the impossible became inevitable. These underwater oases represented something unprecedented on the young Earth's surface. While the rest of the planet struggled under a toxic atmosphere and received only weak energy from a dimmer sun, hydrothermal vents created their own rules. Here, chemical energy flowed in abundance as hot, alkaline water from deep within the Earth met the cooler, more acidic seawater of the primitive oceans. The collision of these different water masses generated steep gradients in temperature, pressure, and chemistry that could drive reactions impossible anywhere else. Scientists studying modern hydrothermal vents have discovered something remarkable. These environments don't just support life, they create the conditions where life's fundamental chemistry can spontaneously emerge from non-living matter. Recent experiments have shown that the extreme pressures and unique chemical conditions found at these ancient underwater vents could facilitate the formation of organic molecules essential for life. Carbon and hydrogen atoms that would normally never combine suddenly find themselves pushed together by forces that dwarf anything available on the surface. But pressure alone wasn't enough. The real magic happened in the details of hydrothermal vent chemistry. Two distinct types of vents dominated the ancient seafloor, each offering its own pathway toward biological complexity. The first type, the black smokers, erupted from magma chambers beneath spreading ocean ridges, belching forth superheated water loaded with metals and sulfur compounds. These dramatic formations created intense chemical gradients, and provided abundant energy for reactions that could build complex molecules from simple precursors. Even more intriguing were the alkaline vents, cooler formations where a process called serpentinization generated steady streams of hydrogen gas and created water with a high pH. These gentler systems produced conditions remarkably similar to those found inside modern living cells. The natural pH gradients that formed when alkaline vent water mixed with more acidic seawater mirrored the proton gradients that cells use to generate energy today. It was as if the Earth had spontaneously created biological batteries, storing and releasing chemical energy in patterns that would later become the foundation of cellular metabolism. The serpentinization process itself represents one of nature's most elegant solutions to the energy problem that faces any emerging life. As seawater reacted with iron-rich rocks in the Earth's crust, it generated not only hydrogen gas, but also methane and other simple organic compounds. These reactions had been operating since liquid water first appeared on Earth, creating a steady supply of the chemical building blocks that life would eventually learn to use and modify. Research teams have successfully recreated these ancient conditions in laboratory settings, watching in fascination as simple organic molecules spontaneously organize themselves into more complex structures. Under the right combination of heat, pressure, and chemical gradients, amino acids link together into protein-like chains. Fatty acids assemble into membrane-like sheets that can trap and concentrate other molecules. Nucleotides, the building blocks of genetic material, form chains that begin to show the first hints of information, storage, and replication. But the most significant breakthrough came when scientists created protocells under these hydrothermal conditions. These simple membrane-bound compartments represent the most basic form of cellular organization, consisting of just a lipid boundary surrounding an aqueous solution. Previous attempts to create protocells had failed under the harsh conditions of simulated ancient vents, leading some researchers to propose that life must have emerged in gentler environments, like shallow tide pools or underground springs. The new experiments changed everything. Not only could protocells form under hydrothermal conditions, but heat and alkalinity weren't just acceptable for their formation, they were necessary. The extreme environment that seemed so hostile to life was actually essential for the chemical processes that could bridge the gap between complex chemistry and simple biology. The protocells that formed under these conditions showed remarkable stability and the ability to concentrate organic molecules from their surroundings, creating the kind of controlled chemical environment that could support more complex reactions. These laboratory successes provided crucial support for theories about life's hydrothermal origins, but they also revealed just how sophisticated the chemistry of early life must have been. The formation of even the simplest protocells required precise coordination between multiple chemical processes. Membranes had to form and maintain their integrity while remaining permeable to essential nutrients. Internal chemistry had to develop the ability to process and concentrate important molecules while excluding harmful ones. Most critically, some mechanism had to emerge for storing and copying information that could be passed from one generation to the next. The solution to these challenges lay in the unique properties of hydrothermal vent environments. The constant flow of chemical energy provided fuel for reactions that could maintain complex molecular systems far from chemical equilibrium. The temperature and pressure gradients created natural selection pressures that favored more stable and efficient molecular arrangements. The mineral surfaces present at vent sites could serve as templates and catalysts, helping to organize chemical reactions and concentrate important compounds. Modern studies of hydrothermal vent chemistry have revealed just how rich these environments are in the precursor molecules needed for life. Amino acids, the building blocks of proteins, form readily under vent conditions. Nucleotide bases, essential components of DNA and RNA, can be synthesized from simple starting materials available in vent fluids. Fatty acids and other lipid molecules that could form primitive cell membranes are produced through reactions between carbon dioxide and hydrogen gas. Perhaps most importantly, these reactions don't just produce random mixtures of organic compounds. The specific conditions at hydrothermal vents favor the formation of molecules that show at least some of the properties we associate with living systems. They tend to be more organized, more stable, and more capable of participating in complex chemical networks than the products of most other prebiotic synthesis reactions. The minerals present at hydrothermal vents played a crucial role in organizing these early chemical systems. Iron-sulfur compounds abundant in vent environments could serve as primitive catalysts, speeding up reactions and helping to channel chemical energy into useful pathways. Clay minerals and other crystalline surfaces provided templates where organic molecules could align in ordered arrangements, setting the stage for the kind of molecular organization that characterizes living cells. Some of these mineral catalysts showed properties that were remarkably similar to the metal-containing enzymes that drive metabolism in modern organisms. Iron-sulfur clusters, in particular, remain essential components of many cellular processes today. From photosynthesis to DNA replication, the similarity between ancient mineral catalysts and modern biological enzymes suggests a direct evolutionary connection, with life gradually replacing inorganic catalysts with more sophisticated organic ones, while maintaining the same basic chemical logic. The energy available at hydrothermal vents wasn't just abundant, it was organized in ways that could support the kind of complex, coordinated chemistry that life requires. Unlike the random energy inputs available from lightning strikes or ultraviolet radiation, hydrothermal systems provided steady, predictable flows of chemical energy that could be harnessed by molecular systems capable of capturing and channeling that energy into useful work. This organized energy flow was essential for maintaining the chemical systems that would eventually become living cells. Life requires the ability to create and maintain complex molecular structures that wouldn't exist under normal chemical equilibrium conditions. This demands a constant input of energy, but not just any energy. The energy has to be available in forms that can be captured and used by molecular machinery, and it has to be delivered in ways that don't destroy the very structures it's meant to maintain. Hydrothermal vents provided exactly this kind of organized energy input. The chemical gradients that formed when hot, alkaline vent water mixed with cooler seawater created natural fuel cells that could drive chemical reactions in controlled, predictable ways. The steady flow of reduced compounds from the Earth's interior provided a reliable source of chemical energy that could be tapped by any molecular system capable of facilitating the right kinds of reactions. The transition from complex chemistry to true biology required one more crucial innovation. The systems that emerged from hydrothermal chemistry had to develop the ability to reproduce themselves, creating copies that could inherit both the molecular machinery and the information needed to maintain and improve upon their chemical organization. This step, from complex but ultimately temporary chemical systems to self-replicating entities capable of evolution, represents the true origin of life as we understand it. The evidence suggests this transition happened gradually, with increasingly sophisticated chemical systems emerging from the rich environments around hydrothermal vents. Simple autocatalytic reactions, where the products of a reaction help to catalyze more of the same reaction, could create primitive forms of chemical reproduction. Networks of such reactions could develop increasing complexity and stability, eventually reaching the point where they could maintain themselves and compete with other chemical systems for resources and energy. The fossil record of this transition remains largely hidden, erased by billions of years of geological change and the microscopic scale of the earliest life forms. But the chemical signatures preserved in ancient rocks tell a story of increasing biological complexity and organization. By 3.5 billion years ago, these signatures indicate that sophisticated microbial ecosystems were already established, suggesting that the transition from chemistry to biology had been completed hundreds of millions of years earlier. The success of these early hydrothermal life forms set the stage for one of the most important expansions in the history of biology. As these primitive organisms evolved and diversified, they began to explore new environments and develop new metabolic capabilities. Some learned to thrive in the cooler waters away from vent sites. Others developed the ability to use sunlight as an energy source, setting the stage for the photosynthetic revolution that would eventually transform Earth's atmosphere and create the oxygen-rich world we know today. But that transformation was still far in the future. For now, in the depths of ancient oceans, around the glowing chimneys of underwater volcanoes, life had taken its first tentative steps. The chemical innovations that emerged in these extreme environments would provide the foundation for all subsequent evolution, establishing the basic principles of cellular organization and metabolism that continue to operate in every living thing on Earth today. The story of life's emergence wasn't complete with just the formation of primitive cells around hydrothermal vents. These early organisms faced an entirely different challenge: survival and expansion in a world that offered no roadmap for biological success. The transition from isolated chemical systems to thriving microbial communities required innovations that would fundamentally reshape how life operated on Earth. The earliest prokaryotic cells were metabolic pioneers, developing biochemical pathways that seem almost alien when compared to modern biology. While today's organisms depend heavily on oxygen for their cellular processes, these ancient microbes had to extract energy from a completely oxygen-free environment. They learned to process sulfur compounds, methane, and iron minerals, turning the toxic chemistry of early Earth into fuel for growth and reproduction. Some of these metabolic innovations were so successful that they remain virtually unchanged in certain organisms today. Deep-sea bacteria still use many of the same chemical pathways that were pioneered billions of years ago around hydrothermal vents. The iron-sulfur chemistry that powered the earliest cells continues to operate in the mitochondria of every complex organism on Earth, including the cells in your own body as you watch this video. But the real breakthrough came when these early organisms began to organize themselves into communities. Individual cells, no matter how sophisticated their metabolism, were limited by their size and the resources they could access directly. Cooperation changed everything. Microbial mats, those layered sheets of different bacterial species, represented one of biology's first great social experiments. These weren't random collections of cells thrown together by chance. They were structured communities where different species occupied specific positions based on their metabolic needs and environmental tolerances. The organisms near the surface might specialize in processing whatever sunlight could penetrate the murky atmosphere. Deeper layers housed species that thrived on the waste products of their neighbors, creating efficient recycling systems that maximize the use of available resources. The formation of these microbial communities required a level of biochemical sophistication that's easy to underestimate. Cells had to develop the ability to communicate with their neighbors, coordinating their activities in ways that benefited the entire community. They produced sticky substances that helped bind the community together, creating physical structures that could persist, even as individual cells died and were replaced. Perhaps most remarkably, these communities could modify their local environment in ways that made conditions more favorable for the group as a whole. They altered water chemistry, created localized pH gradients, and even influenced the deposition of minerals around their colonies. In essence, these microscopic organisms were engaging in primitive ecosystem engineering, reshaping their surroundings to better suit their needs. The preservation of these ancient communities in the fossil record represents one of paleontology's greatest achievements. The stromatolites found in Western Australia and South Africa aren't just rock formations. They're architectural records of how these early organisms lived and grew. Each layer tells a story of daily and seasonal cycles, environmental changes, and the responses of biological communities to shifting conditions over thousands of years. The process that created these fossils required extraordinary precision in timing and chemistry. As microbial mats grew and trapped sediment, minerals had to precipitate in just the right way to preserve the delicate structures before they could decay. The chemistry of the surrounding water had to remain stable long enough for mineralization to occur, but not so stable that the biological signals were lost in purely geological processes. Scientists studying these ancient fossils have developed increasingly sophisticated techniques for distinguishing biological from non-biological structures. They examine the microscopic details of mineral formation, looking for patterns that could only have been created by living organisms. They analyze the chemical composition of different layers, searching for isotopic signatures that reveal the metabolic processes of ancient microbes. The carbon isotope ratios preserved in these rocks provide particularly compelling evidence for ancient life. Living organisms preferentially use lighter carbon isotopes in their metabolic processes, creating distinctive chemical signatures that can persist even when physical structures are destroyed. These signatures in rocks over 3 billion years old match the patterns produced by modern microbial communities, providing strong evidence that similar biological processes were operating in the distant past. But isotope analysis reveals more than just the presence of ancient life. Different metabolic pathways produce different isotopic signatures, allowing scientists to reconstruct the types of chemical processes that were operating in ancient microbial communities. The data suggests that by 3.5 billion years ago, prokaryotic organisms had already developed a remarkable diversity of metabolic strategies. Some of these early organisms were already experimenting with primitive forms of photosynthesis, using sunlight to drive chemical reactions that could produce organic compounds from carbon dioxide and water. This wasn't the oxygen-producing photosynthesis that would later transform Earth's atmosphere, but rather simpler systems that used hydrogen sulfide or other compounds as electron donors instead of water. The development of photosynthesis represented a crucial step in life's expansion beyond the limited environments around hydrothermal vents. For the first time, organisms could tap into the vast energy resources of sunlight, freeing them from dependence on the chemical energy available in specialized geological settings. This opened up entirely new habitats, from shallow coastal waters to inland lakes and rivers. The earliest photosynthetic organisms were probably purple and green bacteria that used bacteria chlorophyll to capture light energy. These organisms could thrive in the low oxygen conditions of early Earth while still taking advantage of solar energy for their metabolic needs. They established themselves in shallow water environments where sufficient light could penetrate, creating some of Earth's first truly solar-powered ecosystems. The expansion of photosynthetic communities had profound implications for global biogeochemical cycles. These organisms began removing carbon dioxide from the atmosphere and oceans, incorporating it into organic compounds that could be buried in sediments or used by other organisms. This biological carbon pump represented the beginning of life's role in regulating Earth's climate and atmospheric composition. At the same time, early photosynthetic bacteria were producing organic compounds that could serve as food sources for other organisms. This created the possibility of more complex ecological relationships, with some organisms specializing in primary production through photosynthesis, while others evolved to consume and break down organic matter. The foundation of modern food webs was beginning to take shape in these ancient microbial communities. The success of these early photosynthetic organisms can be seen in the geological record through the formation of banded iron formations, distinctive rock layers that formed when dissolved iron in ancient oceans reacted with small amounts of oxygen produced by primitive photosynthesis. These formations, found in rocks between 2 and 3 billion years old, represent some of the earliest evidence for biological oxygen production on Earth. But the most revolutionary development was still to come. Somewhere around 3 billion years ago, a group of organisms that would later be called cyanobacteria evolved a form of photosynthesis that used water as an electron donor, splitting water molecules and releasing oxygen as a waste product. This innovation would eventually lead to one of the most dramatic transformations in Earth's history, but its immediate effects were more modest. Early oxygen-producing photosynthesis probably developed in specialized microbial mats, where the organisms were partially protected from the toxic effects of the oxygen they were producing. Oxygen was actually poisonous to most of the life forms that had evolved in the oxygen-free conditions of early Earth. The organisms that first began producing oxygen had to simultaneously evolve mechanisms to protect themselves from their own waste product. The chemical innovations required for oxygenic photosynthesis were extraordinarily complex. The process involves splitting water molecules, one of the most chemically stable compounds in nature, and using the liberated electrons to reduce carbon dioxide into organic compounds. This requires sophisticated molecular machinery, capable of handling highly reactive intermediates, while maintaining precise control over electron flow. The evolution of this system represented one of biology's greatest achievements, comparable to the original emergence of life itself. It required the coordinated evolution of multiple protein complexes, sophisticated light harvesting systems, and mechanisms for managing the dangerous reactive oxygen species that are inevitable byproducts of the process. The organisms that accomplished this feat were the ancestors of modern cyanobacteria, and their evolutionary success would eventually reshape the entire planet. But in the beginning, their impact was limited to localized environments where they could maintain the delicate balance between oxygen production and oxygen tolerance that their metabolism required. These early cyanobacteria formed distinctive communities that can still be recognized in the fossil record. Their stromatolites show characteristic patterns of growth and mineral deposition that reflect the unique chemistry of oxygen-producing photosynthesis. The laminated structures they created provide detailed records of daily and seasonal cycles, revealing how these organisms responded to changes in light availability and water chemistry. The preservation of cyanobacterial fossils has provided remarkable insights into the physiology and ecology of these ancient organisms. Microscopic examination reveals cellular structures that are remarkably similar to those of modern cyanobacteria, suggesting that the basic organization of these organisms has remained relatively stable for billions of years. Chemical analysis of ancient cyanobacterial fossils has revealed evidence for some of the same pigments and metabolic compounds found in modern species. The molecular machinery of photosynthesis appears to have been highly conserved throughout evolutionary history, with the same basic chemical processes operating in organisms separated by billions of years of evolution. The success of cyanobacteria in ancient environments was partly due to their ability to form complex, structured communities that could persist through environmental changes. These organisms developed sophisticated mechanisms for coordinating their activities, including chemical communication systems that allowed them to respond collectively to changes in environmental conditions. Cyanobacterial communities also showed remarkable resilience in the face of environmental stress. They could survive periods of drought by forming resistant spores, tolerate extreme temperatures and chemical conditions, and rapidly recolonize habitats after disturbances. These survival strategies allowed them to expand into a wide range of environments and maintain their populations through the dramatic environmental changes that characterized early Earth. The metabolic versatility of early cyanobacteria was crucial to their ecological success. While they are best known for oxygenic photosynthesis, many species could also perform other types of metabolism when light was not available. Some could fix nitrogen, converting atmospheric nitrogen gas into compounds that could be used by other organisms. Others could break down organic matter, playing important roles in nutrient cycling within their communities. This metabolic flexibility allowed cyanobacterial communities to thrive in environments that experience significant variations in light, temperature, and chemical conditions. They could switch between different metabolic strategies depending on environmental circumstances, maintaining their populations and communities even when conditions became challenging for any single metabolic pathway. The ecological relationships within these ancient cyanobacterial communities were surprisingly complex. Different species occupied distinct niches within the community structure, with some specializing in light capture and primary production, while others focused on nutrient cycling or protection from environmental stress. These early examples of ecological specialization presaged the much more complex ecosystems that would evolve later in Earth's history. The waste products of cyanobacterial metabolism became important resources for other organisms in their communities. The oxygen they produced, while initially toxic to many organisms, eventually became a crucial resource for new types of metabolism that could use oxygen to extract energy from organic compounds much more efficiently than anaerobic processes. The organic compounds produced by cyanobacterial photosynthesis provided food sources for heterotrophic organisms that specialized in breaking down and recycling biological materials. This created some of Earth's first true producer-consumer relationships, establishing ecological patterns that continue to operate in modern ecosystems. The success and expansion of cyanobacterial communities set the stage for one of the most important transitions in Earth's history. As these oxygen-producing organisms spread to new environments and increased in abundance, they began to have measurable effects on global atmospheric and oceanic chemistry. The stage was being set for a transformation that would make complex, oxygen-breathing life possible, but that revolution was still hundreds of millions of years in the future. For now, in the shallow seas and tidal environments of the Middle Archean period, cyanobacteria were establishing the principles of ecosystem organization and biogeochemical cycling that would provide the foundation for all subsequent evolution. Their innovations in photosynthesis, community organization, and environmental modification created new possibilities for life on Earth, opening evolutionary pathways that would eventually lead to the incredible diversity of organisms we see today. The chemical fingerprints left by these ancient cyanobacterial communities tell a story that goes far beyond simple survival. As these organisms spread across the shallow seas of the Archean world, they began to participate in global chemical cycles that would fundamentally alter the planet's future. The carbon they fixed from atmospheric carbon dioxide became incorporated into sediments and rocks, creating some of Earth's earliest organic carbon deposits. These buried organic compounds represent a massive transfer of carbon from the atmosphere to the Earth's crust, beginning a process that continues to influence global climate today. The nitrogen-fixing abilities of many cyanobacteria introduced another crucial element into biological systems. Nitrogen is essential for making proteins and nucleic acids, but atmospheric nitrogen gas is largely inaccessible to most organisms. The evolution of nitrogen fixation allowed these early communities to tap into the vast reservoir of atmospheric nitrogen, making this essential element available to other organisms for the first time. This innovation broke one of the fundamental limitations that had constrained early life and opened new possibilities for biological complexity. The phosphorus cycling that occurred within cyanobacterial mats was equally important for the development of more complex biological systems. Phosphorus is essential for energy storage and transfer in living cells, forming the backbone of molecules like ATP that power cellular processes. Early cyanobacterial communities developed efficient mechanisms for capturing and concentrating phosphorus from their environment, creating localized hotspots of this crucial element that could support more diverse microbial ecosystems. But perhaps the most significant innovation of these ancient communities was their development of complex chemical communication systems. Modern research has revealed that cyanobacteria produce a diverse array of chemical signals that allow them to coordinate their activities across large distances. These molecular messages can trigger synchronized responses to environmental changes, coordinate reproductive cycles, and even allow different species to work together in mutually beneficial relationships. The sophistication of these communication systems in 3 billion-year-old communities suggests that the evolution of biological cooperation began much earlier than previously thought. These weren't just random collections of cells competing for resources. They were organized societies with division of labor, coordinated responses to environmental challenges, and mechanisms for maintaining community stability over long periods of time. The physical structures created by these communities were equally impressive. The stromatolites they built weren't just passive accumulations of sediment and minerals. They were actively constructed architectures that created favorable microenvironments for different types of organisms. The layered structure of these formations provided gradients of light, oxygen, nutrients, and pH that different species could exploit according to their specific metabolic requirements. Some of these ancient stromatolites reached heights of several meters, creating complex three-dimensional habitats that supported remarkable diversity within what appeared to be simple microbial communities. The internal structure of these formations reveals sophisticated patterns of growth and mineral deposition that reflect the coordinated activities of multiple species working together to build and maintain their shared living space. The construction process itself required precise timing and coordination between biological and geological processes. The organisms had to produce the right types and amounts of organic compounds to bind sediment particles together. They had to control the pH and chemistry of their local environment to promote the precipitation of minerals that would preserve their structures. And they had to do all of this while maintaining the delicate balance of nutrients and energy that kept their communities alive and growing. The preservation of these structures in the fossil record provides us with unprecedented detail about how these ancient ecosystems functioned. Individual laminations within stromatolites can be traced across entire formations, revealing how communities responded to seasonal changes, environmental disturbances, and long-term shifts in climate and ocean chemistry. Some layers show evidence of rapid growth during favorable conditions, while others reveal periods of stress or dormancy when communities had to survive harsh environmental changes. The microscopic analysis of these fossil communities has revealed cellular structures and arrangements that mirror patterns seen in modern microbial mats. Individual cells, cell divisions, and even specialized structures like gas vesicles that helped ancient cyanobacteria control their position in the water column can still be observed in exceptionally well-preserved specimens. These microscopic details provide direct evidence for the sophisticated cellular biology that had evolved by the Middle Archean period. The chemical analysis of organic compounds preserved within these ancient structures has revealed the presence of complex biomolecules that indicate highly sophisticated metabolic processes. Lipid molecules that form cell membranes, chlorophyll-like compounds involved in photosynthesis, and even traces of proteins and nucleic acids have been identified in rocks over 3 billion years old. These molecular fossils provide direct evidence for the types of biochemical processes that were operating in ancient microbial communities. The isotopic signatures preserved in these organic compounds reveal details about the metabolic pathways that ancient organisms used to process carbon, nitrogen, and sulfur. Different types of metabolism produce characteristic isotopic patterns, allowing scientists to reconstruct the ecological relationships and nutrient cycling processes that operated within ancient microbial communities. The data shows that by 3 billion years ago, these communities had developed metabolic complexity that rivals what we see in modern ecosystems. The geographic distribution of these ancient fossil communities provides insights into the environmental conditions that existed during the Middle Archean period. Stromatolite deposits from this time period have been found on multiple continents, suggesting that suitable environments for complex microbial life were widespread. The characteristics of different fossil localities reveal variations in water depth, salinity, temperature, and nutrient availability that influenced how these ancient ecosystems developed. Some of the most spectacular examples come from shallow marine environments, where tidal action created regular cycles of exposure and submersion. These dynamic conditions favored the development of particularly complex community structures, with different zones adapted to different phases of the tidal cycle. The organisms living in the highest zones had to tolerate periodic desiccation and intense solar radiation, while those in deeper zones faced different challenges related to light limitation and nutrient availability. The seasonal patterns preserved in these ancient structures reveal that Earth's climate 3 billion years ago was already showing regular cyclical variations. Layers within stromatolites show evidence for seasonal changes in temperature, precipitation, and nutrient availability that influenced how microbial communities grew and reproduced. These patterns suggest that despite the alien chemistry of the Archean atmosphere, Earth's climate system was already developing the regular patterns that would later support more complex forms of life. The evidence for periodic disturbances preserved in the fossil record shows that these ancient ecosystems were remarkably resilient in the face of environmental challenges. Layers that record sudden changes in water chemistry, sediment deposition, or community composition are often followed by evidence for rapid recovery and reorganization. The organisms that built these communities had evolved sophisticated strategies for surviving and recovering from environmental stress that would have been devastating to less organized biological systems. The transition zones between different types of stromatolite structures provide evidence for the environmental gradients that existed in ancient marine environments. Areas where deep water communities graded into shallow water ones show systematic changes in community structure and mineral composition that reflect the different environmental conditions and biological processes operating at different depths. These transition zones were often sites of particularly high biological diversity, with specialized organisms adapted to the unique conditions that existed at environmental boundaries. The role of trace elements in these ancient ecosystems reveals the sophisticated biogeochemical cycling that had evolved by the Middle Archean period. Elements like iron, manganese, and zinc show distinctive patterns of concentration and depletion that reflect their biological processing by ancient microbial communities. Some organisms had evolved mechanisms for concentrating specific trace elements from very dilute solutions, creating localized deposits that could be used by other members of the community. The evolution of metal tolerance and processing capabilities in these ancient organisms was crucial for life's expansion into environments that contained high concentrations of potentially toxic elements. The early Earth's oceans contained much higher levels of dissolved metals than modern seawater, creating challenges that required sophisticated detoxification and metal handling mechanisms. The organisms that successfully colonized these environments developed biochemical innovations that would later prove essential for more complex forms of life. The symbiotic relationships that developed within these ancient microbial communities presaged the more complex cooperative relationships that would evolve later in Earth's history. Evidence suggests that different species within stromatolite communities had evolved complementary metabolic capabilities that allowed them to share resources and waste products in mutually beneficial ways. These early examples of biological cooperation established patterns of interaction that would become increasingly important as life evolved greater complexity. The development of specialized cellular structures within these ancient communities shows that even prokaryotic organisms were capable of remarkable morphological innovation. Some cells developed complex internal membrane systems that allowed them to compartmentalize different metabolic processes. Others evolved specialized appendages that helped them attach to surfaces or move through their environment. These structural innovations increased the efficiency of cellular processes and allowed organisms to exploit new ecological niches. The evidence for horizontal gene transfer between different species within these ancient communities reveals that genetic innovation was occurring much more rapidly than would be possible through simple vertical inheritance. Organisms were sharing genetic material across species boundaries, allowing beneficial mutations and metabolic innovations to spread quickly through entire communities. This process accelerated the evolution of new capabilities and helped ancient microbial communities respond rapidly to environmental challenges. The seasonal reproduction cycles preserved in the fossil record show that these ancient organisms had evolved sophisticated mechanisms for timing their reproductive activities. Some species synchronize their reproduction with seasonal changes in light availability, nutrient levels, or water chemistry. Others showed evidence for complex life cycles that involved different morphological forms adapted to different environmental conditions or life stages. The preservation of ancient microbial communities in multiple different types of environments reveals the remarkable adaptability that prokaryotic life had achieved by the Middle Archean period. Communities have been found in marine environments, ranging from deep ocean basins to shallow tidal flats, in freshwater lakes and rivers, and even in terrestrial environments where water was available seasonally. This geographic and environmental diversity shows that microbial life had already begun to explore most of the habitable environments available on early Earth. The chemical innovations that evolved within these ancient communities established many of the fundamental biochemical pathways that continue to operate in modern organisms. The electron transport chains that power cellular respiration, the carbon fixation pathways that incorporate atmospheric carbon dioxide into organic compounds, and the nitrogen fixation systems that make atmospheric nitrogen available to biological systems all trace their origins to innovations that first appeared in Archean microbial communities. The regulatory mechanisms that allowed ancient organisms to coordinate their metabolic activities in response to environmental changes show remarkable sophistication for such apparently simple life forms. These organisms had evolved complex genetic switches that could turn different metabolic pathways on or off, depending on the availability of nutrients, light, or other environmental factors. They could rapidly adjust their cellular processes to take advantage of favorable conditions or survive periods of environmental stress. The development of resistance mechanisms that allowed ancient organisms to survive extreme environmental conditions established the foundation for life's later expansion into even more challenging environments. Some organisms evolved the ability to form resistant spores that could survive complete desiccation, extreme temperatures, or exposure to high levels of radiation. Others developed sophisticated repair mechanisms that could fix damage to their genetic material or cellular structures caused by environmental stress. The emergence of primitive immune systems in these ancient communities shows that biological conflict and cooperation were already important evolutionary forces 3 billion years ago. Some organisms evolved mechanisms for recognizing and defending against harmful microbes, while others developed strategies for avoiding or overcoming these defensive systems. These early biological arms races drove rapid evolution and innovation within microbial communities. The evidence for primitive photosynthetic systems in these ancient communities reveals the gradual evolution of the complex molecular machinery that would eventually make oxygenic photosynthesis possible. Early photosynthetic organisms used simpler systems that could capture light energy and use it to drive chemical reactions. But these systems were much less efficient than modern photosynthesis and produced different waste products. The evolution of increasingly sophisticated light harvesting and energy conversion systems set the stage for the revolutionary innovations that would come later. The biogeochemical cycles that operated within these ancient communities were already showing many of the characteristics that define modern ecosystems. Primary producers converted inorganic compounds into organic matter using energy from light or chemical reactions. Decomposer organisms broke down organic matter and recycled nutrients back into forms that could be used by primary producers. Predatory microbes consumed other organisms and transferred energy through primitive food webs. These fundamental ecological relationships had been established well before more complex forms of life appeared on Earth. But the most profound impact of these ancient microbial communities was yet to come. The oxygen that cyanobacteria had begun producing as a waste product of their photosynthetic activities was starting to accumulate in local environments around their colonies. Initially, this oxygen was quickly consumed by reactions with dissolved iron and other chemicals in the oceans and atmosphere. But as cyanobacterial communities expanded and became more productive, they began to produce oxygen faster than it could be consumed by these chemical sinks. The stage was being set for one of the most dramatic transformations in Earth's history, an event that would poison most existing life forms while simultaneously creating the conditions necessary for entirely new types of organisms to evolve. The Great Oxidation Event was still hundreds of millions of years in the future, but the biological innovations that would drive it were already taking shape in the shallow seas and tidal environments of the Middle Archean world. The success of these ancient cyanobacterial communities had established life as a major force in Earth's environmental systems. No longer were biological processes confined to specialized environments like hydrothermal vents or isolated chemical systems. Life had become a global phenomenon capable of influencing atmospheric composition, ocean chemistry, and even the geological processes that shaped the planet's surface. The microscopic organisms thriving in ancient stromatolites were about to change the world. The transformation wouldn't happen overnight. For hundreds of millions of years, the oxygen produced by these ancient cyanobacterial communities remained trapped in local environments, quickly consumed by iron and sulfur compounds that were abundant in Archean oceans. But something remarkable was happening beneath the surface of this chemical balance. The biological machinery that these organisms had developed was becoming more efficient, more productive, and more widespread with each passing generation. The evolutionary pressure on these early oxygen producers was intense and unforgiving. Oxygen was literally toxic to most life forms that had evolved in the oxygen-free conditions of early Earth. The organisms producing it had to simultaneously evolve sophisticated defense mechanisms against their own waste product. This created a biological arms race between oxygen production and oxygen tolerance that would drive some of the most important innovations in cellular biology. The solution came through the evolution of antioxidant systems, complex molecular machinery that could neutralize the damaging effects of oxygen and its reactive derivatives. These systems used specialized enzymes and chemical compounds to convert harmful oxygen species into less dangerous forms, protecting cellular components from oxidative damage. The organisms that successfully developed these protective mechanisms gained a crucial advantage over their competitors. But the real breakthrough came when some organisms learned to turn the tables on oxygen entirely. Instead of simply tolerating this toxic waste product, they evolved metabolic pathways that could actually use oxygen as a tool for extracting energy from organic compounds. Aerobic respiration, the process that powers most modern life forms, began as an adaptation to survive in an increasingly oxygen-rich world. The efficiency gains from aerobic metabolism were staggering. Organisms that could use oxygen to break down organic compounds could extract roughly 18 times more energy from the same amount of food compared to anaerobic processes. This metabolic revolution provided the energy budget that would eventually make complex multicellular life possible. But that development was still billions of years in the future. The chemical evidence for this metabolic transition is preserved in rock formations that date to around 2.5 billion years ago. Banded iron formations, those distinctive striped rocks that formed when dissolved iron reacted with oxygen in ancient oceans, suddenly become much more abundant in the geological record. These formations represent a massive chemical experiment conducted on a planetary scale, as the iron-rich oceans of early Earth gradually became oxidized. The process wasn't smooth or gradual. The geological record reveals that early oxygen production occurred in pulses and cycles, with periods of increased oxygen production followed by intervals when oxygen levels dropped again. This irregular pattern reflects the complex interactions between biological oxygen production, chemical oxygen consumption, and the physical processes that mixed oxygen through the ocean atmosphere system. The organisms driving this transformation were developing increasingly sophisticated strategies for managing their oxygen-rich environment. Some species evolved the ability to migrate vertically through the water column, moving to deeper, more oxygen-poor layers when surface conditions became too harsh. Others developed complex life cycles that included resistant stages, capable of surviving extreme oxygen exposure. The community structures that these organisms built became increasingly elaborate as they adapted to the changing chemical environment. Stromatolites from this period show more complex internal architectures, with specialized zones for different types of metabolism and more sophisticated systems for managing the flow of nutrients and waste products. These structures became biological factories for oxygen production, processing vast quantities of carbon dioxide and water while releasing oxygen into their surroundings. The geographic expansion of these oxygen-producing communities was remarkable. Fossil evidence shows that cyanobacterial stromatolites spread to virtually every marine environment where sufficient light was available for photosynthesis. From tropical shallow seas to polar regions, these organisms established thriving colonies that began the slow process of transforming Earth's atmospheric composition. The impact on other forms of life was initially catastrophic. Most of the anaerobic organisms that had dominated Earth's biosphere for over 1 billion years were highly sensitive to oxygen toxicity. Mass extinction events during this period eliminated many ancient lineages, creating ecological niches that would be filled by new types of organisms better adapted to oxygen-rich conditions. But extinction wasn't the only response to rising oxygen levels. Some anaerobic organisms survived by retreating to oxygen-free environments like deep ocean sediments, underground aquifers, and the anoxic zones that persisted in some marine basins. These refugia became evolutionary laboratories where ancient metabolic pathways were preserved and continued to evolve, maintaining biological connections to Earth's oxygen-free past. The organisms that thrived in the new oxygen-rich world began to explore metabolic possibilities that had been impossible under anaerobic conditions. The energy abundance provided by aerobic respiration supported the evolution of larger, more complex cellular structures and more sophisticated behavioral capabilities. Some organisms developed primitive sensory systems that allowed them to detect and respond to chemical gradients in their environment. The development of cellular motility became increasingly important as organisms competed for resources in the changing environment. Swimming, gliding, and crawling mechanisms allowed microbes to actively seek out favorable conditions and avoid harmful ones. These locomotory innovations required significant energy investments, but provided crucial advantages in environments where resources were patchy and conditions were rapidly changing. The evolution of predatory behavior among microorganisms represented another major innovation during this period. Some species developed the ability to engulf and digest other cells, creating the first true predator-prey relationships in Earth's biosphere. This biological innovation established food webs based on direct consumption rather than just chemical cycling, adding new levels of complexity to ecosystem organization. The molecular machinery required for cellular predation was remarkably sophisticated. Predatory microbes had to evolve mechanisms for recognizing suitable prey, capturing and immobilizing target cells, and breaking down cellular components for nutrition. They also needed defensive systems to protect themselves from becoming prey to other predatory organisms. These biological arms races drove rapid evolution and innovation throughout microbial communities. The waste products of these predatory interactions became important resources for other organisms in the ecosystem. The organic compounds released when cells were digested provided nutrition for decomposer species, while the mineral components of cellular structures contributed to the chemical cycling that maintained ecosystem productivity. These early food webs were establishing the fundamental patterns of energy and material flow that characterize all modern ecosystems. The seasonal and daily cycles that influence these ancient ecosystems were becoming more pronounced as Earth's climate system matured. The fossil record shows evidence for regular patterns of growth and reproduction that were synchronized with environmental cycles. Some organisms developed internal biological clocks that allowed them to anticipate and prepare for predictable environmental changes. The development of dormancy strategies became crucial for surviving periods of environmental stress. Many organisms evolved the ability to enter quiescent states during unfavorable conditions, reducing their metabolic activity to minimal levels and waiting for conditions to improve. These survival strategies allowed communities to persist through environmental disturbances that would otherwise have caused local extinctions. The chemical communication systems that operated within these communities became increasingly sophisticated as organisms developed new ways to coordinate their activities. Some species began producing complex mixtures of signaling molecules that could convey detailed information about environmental conditions, resource availability, and potential threats. These chemical languages allowed communities to respond collectively to challenges that individual organisms couldn't handle alone. The formation of specialized cellular structures for producing and detecting chemical signals required significant evolutionary innovation. Organisms had to develop molecular machinery for synthesizing specific signaling compounds, transport systems for delivering these molecules to target locations, and receptor systems for detecting and responding to chemical messages from other organisms. These communication systems laid the groundwork for the more complex coordination mechanisms that would evolve in multicellular organisms. The genetic mechanisms that controlled these increasingly complex biological processes were becoming more sophisticated as organisms developed new ways to regulate gene expression. Environmental sensing systems allowed cells to detect changes in their surroundings and adjust their metabolic activities accordingly. These regulatory networks provided the flexibility needed to survive in environments that were becoming more dynamic and unpredictable. The horizontal transfer of genetic material between different species remained an important source of evolutionary innovation during this period. Organisms continued to share beneficial mutations and metabolic capabilities across species boundaries, allowing entire communities to adapt rapidly to changing environmental conditions. This genetic cooperation accelerated the evolution of new biological capabilities and helped communities explore new ecological possibilities. The development of more sophisticated DNA repair mechanisms became crucial as organisms faced increasing levels of oxidative stress from rising oxygen concentrations. The reactive oxygen species produced by aerobic metabolism could cause significant damage to genetic material, requiring robust cellular systems for detecting and repairing DNA damage. The organisms that successfully evolved these protective mechanisms gained significant survival advantages. The metabolic flexibility that characterized these ancient organisms allowed them to thrive in environments that were undergoing rapid chemical and physical changes. Many species retained the ability to switch between different metabolic pathways depending on environmental conditions, using aerobic respiration when oxygen was available, but reverting to anaerobic processes when necessary. This metabolic versatility provided crucial insurance against environmental unpredictability. The biogeochemical cycles that operated during this period were becoming increasingly complex as biological processes began to dominate chemical cycling in many environments. The carbon cycle was particularly affected, as photosynthetic organisms removed vast quantities of carbon dioxide from the atmosphere, while aerobic decomposition processes returned organic carbon to the inorganic reservoir. These biological influences on global chemistry were setting the stage for the climate regulation systems that would become crucial for maintaining habitable conditions on Earth. The phosphorus and nitrogen cycles were also becoming more tightly coupled to biological processes, as organisms developed more efficient mechanisms for capturing and recycling these essential nutrients. The evolution of specialized enzymes for processing organic phosphorus compounds allowed communities to access nutrient sources that had previously been unavailable. Similarly, Improvements in nitrogen fixation and recycling processes increased the productivity of ecosystems in nitrogen-limited environments. The sulfur cycle was particularly important during this period because many organisms still depended on sulfur-based metabolism for their energy needs. The interactions between sulfur-reducing and sulfur-oxidizing bacteria created complex chemical gradients that supported diverse microbial communities. These sulfur-based ecosystems provided alternative energy sources that allowed life to thrive in environments where photosynthesis was not possible. The iron cycle was being fundamentally transformed by rising oxygen levels as dissolved iron in the oceans reacted with oxygen to form insoluble iron oxides. This process removed both iron and oxygen from the water column, creating the banded iron formations that are characteristic of this period in Earth's history. The biological communities that depended on iron-based metabolism had to adapt to decreasing iron availability while coping with increasing oxygen toxicity. The trace element cycles that supported these ancient ecosystems were becoming more complex as organisms developed increasingly sophisticated requirements for specific minerals and metals. Some organisms evolved mechanisms for concentrating rare elements from very dilute solutions, creating localized deposits that could support specialized biological processes. These biogeochemical innovations increased the efficiency of ecosystem functioning and allowed communities to thrive in environments with limited nutrient availability. The physical structures created by these biological communities were becoming more elaborate and diverse as organisms experimented with new approaches to ecosystem engineering. Some communities built towering reef-like structures that created complex three-dimensional habitats, supporting remarkable biological diversity. Others developed extensive mat systems that covered large areas of the seafloor and influenced local patterns of sediment deposition and water circulation. The preservation potential of these biological structures was improving as organisms developed better mechanisms for promoting the mineralization processes that created fossils. Some species produced organic compounds that encouraged the precipitation of specific minerals, while others created microenvironments with chemical conditions that favored rapid fossilization. These innovations in biological preservation have provided paleontologists with increasingly detailed records of ancient life. The environmental gradients that existed within these ancient ecosystems were becoming steeper and more pronounced as biological processes created stronger chemical and physical contrasts. The boundaries between oxic and anoxic zones became sites of intense biological activity, with specialized organisms adapted to the unique conditions that existed at these interfaces. These gradient environments were often centers of evolutionary innovation, where new metabolic capabilities and ecological strategies first appeared. The temporal dynamics of these ecosystems were becoming more complex as organisms developed increasingly sophisticated responses to environmental variability. Some communities showed evidence for complex succession patterns, with different species dominating during different phases of ecosystem development. Others displayed cyclic patterns of activity that reflected seasonal or longer-term environmental changes. The resilience mechanisms that allowed these ecosystems to survive major environmental disturbances were becoming more sophisticated as organisms developed better strategies for recovery and reorganization. Some communities evolved seed bank strategies, maintaining dormant populations that could rapidly recolonize disturbed areas. Others developed modular growth patterns that allowed them to survive partial destruction and regrow from surviving fragments. The geographic connectivity between distant ecosystems was increasing as organisms developed better mechanisms for dispersal and colonization. Some species evolved resistant propagules that could survive long-distance transport through hostile environments. Others developed life cycles that included dispersive stages capable of establishing new populations in suitable habitats. These improvements in biological dispersal allowed successful innovations to spread rapidly between different geographic regions. The evolutionary innovations that emerged during this period established many of the fundamental biological processes that continue to operate in modern ecosystems. The oxygen revolution that began with ancient cyanobacteria would eventually create the atmospheric conditions necessary for complex multicellular life. The metabolic pathways that evolved to cope with oxygen toxicity would become the foundation for the high-energy metabolism that powers modern animals. The ecological relationships that develop between different types of microorganisms would provide the blueprint for the complex food webs that characterize modern ecosystems. But perhaps most importantly, this period established life as a dominant force in Earth's environmental systems. The biological processes that operated in these ancient microbial communities were already beginning to influence global atmospheric composition, ocean chemistry, and climate patterns. The microscopic organisms thriving in Archean stromatolites were laying the groundwork for the biosphere that would eventually support the incredible diversity of life we see on Earth today. The stage was set for the next great chapter in life's evolutionary story. The transformation that began in those ancient microbial communities wasn't just biological, it was planetary. As cyanobacteria spread across the shallow seas of the Archean world, they initiated a chemical revolution that would fundamentally alter every aspect of Earth's environmental systems. The oxygen they produced wasn't just accumulating in their immediate surroundings anymore. It was beginning to escape into the broader ocean and atmosphere, setting in motion changes that would rewrite the rules of life on Earth. The process unfolded over hundreds of millions of years, but its effects were anything but gradual when they finally reached critical thresholds. The iron-rich oceans that had characterized early Earth became massive chemical battlegrounds, where dissolved iron met biological oxygen in reactions that painted the seafloor with distinctive red and black stripes. These banded iron formations represent some of the most spectacular evidence of life's growing influence on planetary chemistry. Vast deposits of oxidized iron that formed as ancient cyanobacteria pumped oxygen into iron-saturated waters. But the iron formations tell only part of the story. The real drama was happening in the microbial communities themselves, where organisms were locked in an evolutionary arms race between oxygen production and oxygen tolerance. The cyanobacteria that had started this chemical revolution faced a deadly paradox. The waste product of their own metabolism was becoming toxic to their cellular machinery, forcing them to evolve increasingly sophisticated defenses against the very compound that would eventually make complex life possible. The solutions these organisms developed were nothing short of revolutionary. They evolved antioxidant systems, molecular machinery capable of neutralizing the damaging effects of reactive oxygen species before they could destroy essential cellular components. These weren't simple chemical buffers, but complex enzymatic networks that could detect, intercept, and neutralize oxygen radicals with remarkable precision. The organisms that successfully developed these protective systems gained crucial advantages over their competitors, allowing them to thrive in increasingly oxygen-rich environments. Some of the most ingenious innovations involved compartmentalization strategies that kept oxygen-sensitive processes isolated from oxygen-rich parts of the cell. Ancient cyanobacteria developed internal membrane systems that created specialized zones for different metabolic activities, protecting vulnerable biochemical pathways while maintaining access to the benefits of oxygenic photosynthesis. These cellular architectures represented early experiments in the kind of compartmentalization that would later become essential for eukaryotic cell organization. The competitive dynamics within these evolving microbial communities drove rapid innovation in cellular defense mechanisms. Organisms that could produce more potent antioxidants or develop better oxygen tolerance gained access to higher energy environments where photosynthesis was most efficient. This created positive feedback loops, where success in oxygen-rich conditions led to even greater oxygen production, accelerating the pace of environmental change and evolutionary adaptation. The chemical warfare occurring at the microscopic level had profound implications for community structure and ecological relationships. Some organisms began producing compounds specifically designed to overwhelm the antioxidant defenses of their competitors, using oxygen toxicity as a biological weapon. Others developed cooperative relationships, where different species shared the metabolic burden of oxygen detoxification, creating some of the earliest examples of biochemical mutualism. The arms race between oxygen production and oxygen tolerance wasn't confined to photosynthetic organisms. Every member of these ancient microbial communities had to adapt to the changing chemical environment or face extinction. Many anaerobic species that had dominated early Earth's biosphere found themselves increasingly restricted to oxygen-poor refugia, while others evolved partial oxygen tolerance that allowed them to survive in moderately oxygenated conditions. The evolutionary pressure was so intense that some organisms developed the ability to switch their entire metabolic strategy depending on oxygen availability. These metabolically flexible species could perform oxygenic photosynthesis when conditions were favorable, but revert to anaerobic processes when oxygen levels became too high or when other environmental factors made photosynthesis impractical. This metabolic versatility provided crucial insurance against the unpredictable environmental changes that characterized this transitional period. The success of oxygen-tolerant organisms began to shift the entire trajectory of life on Earth. Communities dominated by these adapted species became more productive and more stable than their anaerobic predecessors, creating positive feedback loops that accelerated the spread of oxygenic photosynthesis. The geographic expansion of cyanobacterial communities meant that oxygen production was no longer confined to isolated environments, but was becoming a global phenomenon. The chemical signatures preserved in rocks from this period reveal the growing influence of biological oxygen production on global biogeochemical cycles. Carbon isotope ratios show evidence for increasing rates of organic carbon burial, suggesting that photosynthetic communities were removing vast quantities of carbon dioxide from the atmosphere. Sulfur isotope patterns indicate changes in the cycling of sulfur compounds as oxygen began to influence sulfur-based metabolic pathways. The nitrogen cycle was also being transformed as organisms developed new strategies for processing nitrogen compounds in oxygen-rich environments. Some species evolved the ability to perform nitrification, using oxygen to convert ammonia into nitrate compounds that could serve as alternative nitrogen sources for other organisms. These innovations increased the efficiency of nitrogen cycling and supported higher levels of biological productivity in nutrient-limited environments. The development of aerobic nitrogen fixation represented another crucial innovation during this period. Traditional nitrogen fixation was highly sensitive to oxygen because the enzymes involved were rapidly inactivated by oxygen exposure. The evolution of protective mechanisms that could shield nitrogen-fixing enzymes from oxygen damage while maintaining access to the energy provided by aerobic metabolism was a remarkable biochemical achievement that expanded the environments where nitrogen fixation could occur. The phosphorus cycle was becoming more tightly coupled to biological processes as organisms developed increasingly efficient mechanisms for capturing and recycling this essential nutrient. Some species evolved the ability to dissolve phosphorus-containing minerals using organic acids, while others developed sophisticated intracellular storage systems that allowed them to accumulate phosphorus during times of abundance and use it during periods of scarcity. The trace element requirements of these evolving communities were becoming more complex as organisms developed new metabolic pathways that required specific metals and minerals. Copper became increasingly important for enzymes involved in oxygen metabolism, while manganese played crucial roles in protecting cells from oxidative damage. The evolution of sophisticated metal handling systems allowed organisms to access these essential elements while avoiding toxicity from excessive accumulation. The physical structures created by these oxygen-adapted communities were becoming increasingly elaborate and diverse. Some stromatolites from this period show evidence for complex internal circulation systems that allowed oxygen-rich water to penetrate deep into the community structure, while maintaining anoxic zones where oxygen-sensitive organisms could survive. These architectural innovations created three-dimensional habitats that could support remarkable biological diversity within relatively small spatial scales. The temporal dynamics of these communities were becoming more pronounced as organisms developed increasingly sophisticated responses to daily and seasonal cycles of oxygen production and consumption. Some species evolved the ability to migrate vertically through the community structure, following optimal oxygen concentrations as they changed throughout the day. Others developed complex life cycles that synchronized reproduction and dispersal with predictable environmental changes. The development of biological rhythms that could track environmental cycles represented a major advance in cellular organization. Organisms evolved internal timing mechanisms that allowed them to anticipate and prepare for predictable changes in oxygen availability, light intensity, and nutrient concentrations. These biological clocks provided significant competitive advantages by allowing organisms to optimize their metabolic activities for changing environmental conditions. The chemical communication systems operating within these communities were becoming more sophisticated as organisms developed new ways to coordinate their responses to oxygen stress and other environmental challenges. Some species began producing signaling molecules that could trigger community-wide responses to sudden changes in oxygen concentration, allowing entire populations to rapidly adjust their metabolic activities when conditions became dangerous. The evolution of quorum-sensing mechanisms allowed organisms to monitor population density and adjust their activities accordingly. High population densities could lead to dangerous oxygen accumulation during peak photosynthetic periods, requiring coordinated responses to prevent community-wide oxygen toxicity. These chemical communication systems provided the foundation for the complex coordination mechanisms that would later evolve in multicellular organisms. The genetic innovations that supported these increasingly complex biological processes were remarkable in their sophistication. Organisms evolved regulatory networks that could rapidly adjust gene expression in response to changing oxygen concentrations, allowing them to fine-tune their metabolic activities for optimal performance under varying environmental conditions. These regulatory systems required precise coordination between multiple genetic switches and feedback mechanisms. The development of stress response systems became crucial for surviving the oxidative challenges of this transitional period. Organisms evolved genetic circuits that could detect cellular damage from oxygen exposure and activate repair mechanisms before the damage became lethal. These stress response systems included sophisticated DNA repair pathways, protein refolding mechanisms, and cellular cleaning systems that could remove damaged cellular components. The horizontal transfer of genetic material remained an important mechanism for sharing adaptive innovations between different species and communities. Genes encoding antioxidant enzymes, oxygen tolerance mechanisms, and stress response systems spread rapidly through microbial populations, allowing beneficial adaptations to be shared across species boundaries. This genetic cooperation accelerated the pace of evolutionary adaptation to changing environmental conditions. The metabolic networks that evolved during this period were becoming increasingly integrated and efficient. Organisms developed biochemical pathways that could channel the energy released by oxygen reactions into useful cellular processes, transforming oxygen from a toxic waste product into a valuable metabolic resource. These early experiments with aerobic metabolism laid the groundwork for the high-energy biochemistry that would later make complex multicellular life possible. The evolution of electron transport chains that could safely handle oxygen represented one of the most important biochemical innovations in the history of life. These molecular machines could extract energy from the controlled reaction of organic compounds with oxygen while preventing the formation of dangerous reactive oxygen species. The development of these sophisticated energy conversion systems required precise coordination between multiple protein complexes and regulatory mechanisms. The cellular membranes that house these complex biochemical systems had to evolve new properties to function effectively in oxygen-rich environments. The lipid composition of cellular membranes changed to provide better protection against oxidative damage while maintaining the permeability properties needed for efficient metabolism. These membrane innovations required the evolution of new biosynthetic pathways for producing specialized lipid molecules. The protein folding mechanisms that operated within these cells had to adapt to the oxidative conditions that could disrupt normal protein structure. Organisms evolved molecular chaperone systems that could help proteins maintain their proper shapes in oxygen-rich environments, as well as quality control mechanisms that could identify and remove proteins that had been damaged by oxidative stress. The energy storage systems used by these organisms were becoming more sophisticated as they developed ways to capture and store the abundant energy available from aerobic metabolism. Some species evolved the ability to produce energy-rich compounds that could be stored during periods of high photosynthetic activity and used during times when energy demands exceeded photosynthetic capacity. The waste management systems operating within these communities were becoming increasingly important as higher metabolic rates produced larger quantities of potentially harmful waste products. Organisms evolved mechanisms for detoxifying and disposing of metabolic wastes that could interfere with cellular processes or harm neighboring organisms. These waste processing systems required sophisticated biochemical pathways and transport mechanisms. The defensive systems that protected these communities from environmental threats were becoming more elaborate as organisms faced increasing challenges from oxidative stress, toxic metals, and other environmental hazards. Some species developed the ability to produce protective compounds that could shield entire communities from harmful environmental conditions, while others evolved rapid response mechanisms that could quickly isolate and contain environmental threats. The reproductive strategies employed by these organisms were adapting to the changing environmental conditions and increased metabolic capabilities. Some species evolved more complex reproductive cycles that could take advantage of the energy abundance provided by aerobic metabolism, while others developed reproductive timing mechanisms that synchronized reproduction with optimal environmental conditions. The dispersal mechanisms used by these organisms were becoming more sophisticated as communities developed better strategies for colonizing new environments and maintaining genetic connectivity between distant populations. Some species evolved resistant propagules that could survive transport through increasingly oxygen-rich environments, while others developed life cycle stages specifically adapted for dispersal and colonization. The symbiotic relationships that developed within these communities were becoming more intimate and specialized as organisms explored new forms of metabolic cooperation. Some partnerships involved the sharing of metabolic products between species with complementary biochemical capabilities, while others involved more integrated relationships where different organisms provided essential services for their partners. The competitive interactions within these communities were intensifying as organisms developed more sophisticated strategies for accessing limited resources and defending against biological threats. Some species evolved the ability to produce antimicrobial compounds that could suppress competitor populations, while others developed resistance mechanisms that protected them from biological attacks. The ecosystem engineering activities of these communities were having increasingly profound effects on their local environments. Some organisms developed the ability to modify water chemistry over large spatial scales, creating environmental conditions that favored their own growth while making conditions difficult for competitors. These biological modifications of environmental conditions represented early examples of niche construction that would become increasingly important in later evolution. The resilience mechanisms that allowed these communities to survive major environmental disturbances were becoming more sophisticated as organisms developed better strategies for recovery and reorganization. Some communities evolved the ability to maintain dormant populations that could rapidly recolonize disturbed areas, while others developed modular growth patterns that allowed them to survive partial destruction and regrow from surviving fragments. The seasonal patterns that characterize these ecosystems were becoming more pronounced as organisms developed increasingly sophisticated responses to environmental variability. Some communities showed evidence for complex succession patterns that reflected seasonal changes in environmental conditions and resource availability. Others displayed cyclical patterns of activity that maximize productivity during favorable periods while minimizing energy expenditure during stressful conditions. The geographic patterns of biodiversity that emerged during this period reflected the complex interactions between environmental gradients, dispersal limitations, and evolutionary processes. Some regions became centers of evolutionary innovation, where new metabolic capabilities and ecological strategies first appeared, while others served as refugia where ancient lineages could persist, despite changing global conditions. The biogeographic connections between distant communities were strengthening as organisms developed better mechanisms for long-distance dispersal and colonization. The spread of successful evolutionary innovations across large geographic distances was accelerating the pace of global evolutionary change and increasing the connectivity of Earth's emerging biosphere. The paleo-environmental records preserved in rocks from this period provide detailed documentation of the environmental changes that accompanied this biological revolution. Geochemical indicators show systematic changes in atmospheric and oceanic chemistry that reflect the growing influence of biological oxygen production on global environmental systems. The isotopic signatures preserved in organic matter from this period reveal increasing sophistication in biological carbon processing and metabolic pathway diversity. Different types of photosynthesis and carbon fixation produce distinctive isotopic signatures, allowing scientists to reconstruct the metabolic diversity that characterized ancient microbial communities. The mineral deposits formed during this period provide evidence for the complex interactions between biological processes and geochemical cycles. The precipitation of specific minerals in association with biological structures indicates that organisms were actively influencing local geochemistry and creating conditions that favored the preservation of their remains. The trace fossil evidence from this period shows increasing behavioral complexity and environmental modification by ancient organisms. Burrow structures, feeding traces, and other evidence for biological activity indicate that organisms were developing increasingly sophisticated strategies for exploiting their environments and modifying local conditions. The molecular fossils preserved in rocks from this period provide direct evidence for the biochemical innovations that characterize this evolutionary transition. Organic compounds that are specific to particular metabolic pathways or cellular structures can be identified in ancient rocks, providing insights into the physiological capabilities of extinct organisms. The evolutionary relationships between ancient and modern organisms can be reconstructed using molecular phylogenetic techniques that compare genetic sequences from living species. These analyses reveal that many of the fundamental biochemical innovations that evolved during the oxygen transition are still present in modern organisms, demonstrating the evolutionary continuity between ancient and contemporary life. The experimental studies that recreate ancient environmental conditions in laboratory settings provide insights into the chemical and physical processes that operated during this crucial period in Earth's history. These experiments help scientists understand how biological innovations could have emerged from purely chemical systems and how early life forms could have survived and thrived under the harsh conditions of early Earth. The comparative studies of modern environments that resemble ancient conditions provide valuable insights into how early ecosystems might have functioned. Modern microbial mats, hydrothermal vent communities, and other extreme environments serve as natural laboratories for understanding the ecological processes that shaped early life on Earth. The implications of this ancient evolutionary transition extend far beyond academic interest in Earth's early history. The biological innovations that emerged during the oxygen revolution established many of the fundamental processes that continue to support life on Earth. Today, understanding how life adapted to the challenges of an oxygen-rich world provides insights into the resilience and adaptability of biological systems that may be relevant for understanding how life might respond to current and future environmental changes. The oxygen revolution also provides important perspectives on the search for life beyond Earth. The environmental conditions that existed during this crucial period in Earth's history may be similar to conditions that exist on other planets or moons in our solar system. Understanding how life emerged and evolved under these conditions could inform strategies for detecting and studying extraterrestrial life. The story of Earth's oxygen revolution demonstrates the profound ways that life and environment interact to shape planetary evolution. The biological innovations that emerged from ancient microbial communities didn't just adapt to environmental change. They created new environmental conditions that opened entirely new evolutionary possibilities. This dynamic relationship between life and planet continues to operate today as biological processes influence global climate, atmospheric composition, and geochemical cycles. The legacy of these ancient oxygen-producing communities extends into every aspect of modern biology. The chloroplasts that power photosynthesis in plants are direct descendants of ancient cyanobacteria that were incorporated into eukaryotic cells through endosymbiosis. The mitochondria that generate energy in animal cells evolved from ancient bacteria that had learned to use oxygen for aerobic respiration. Even the antioxidant systems that protect our cells from oxidative damage trace their origins to the defensive mechanisms that evolved in response to the first oxygen crisis. The atmospheric oxygen that we breathe today is the direct product of billions of years of photosynthetic activity that began in those ancient stromatolite communities. Every breath we take connects us to the evolutionary innovations that emerged from the extreme environments of early Earth, reminding us that the air we depend on was once a deadly poison that drove one of the greatest evolutionary transitions in the history of life. The transformation of Earth's atmosphere from oxygen-free to oxygen-rich didn't happen overnight, but when it reached critical thresholds, the changes were dramatic and irreversible. The stage was being set for evolutionary innovations that would eventually lead to the emergence of complex cells, multicellular organisms, and ultimately the incredible diversity of life that characterizes our modern world. The microscopic pioneers that survived the oxygen revolution had fundamentally altered the trajectory of evolution on Earth, creating the conditions that would make our own existence possible billions of years later. The revolution that began in those ancient cyanobacterial communities was accelerating, but its effects remained largely invisible to any hypothetical observer of early Earth. For hundreds of millions of years, the oxygen these organisms produced was immediately consumed by chemical reactions with iron, sulfur, and organic compounds dissolved in the primitive oceans. It was like filling a bathtub with the drain wide open. No matter how much water you poured in, the level remained constant until something fundamental changed. That change came gradually, then suddenly. Around 2.4 billion years ago, the chemical sinks that had been absorbing biological oxygen began to reach saturation. The dissolved iron in the oceans had been largely converted to insoluble oxides. Sulfur compounds had been oxidized to their limits. Organic matter in the water column had been consumed by aerobic processes. For the first time in Earth's history, oxygen began to accumulate in the atmosphere itself. The transformation didn't happen uniformly across the planet. Some regions experienced dramatic increases in atmospheric oxygen, while others remained relatively unchanged for millions of years longer. Ocean basins that were well connected to surface waters saw rapid oxygenation, while isolated deep water masses retained their ancient anoxic chemistry. This created a patchwork world where organisms faced wildly different environmental conditions depending on their location. The geological evidence for this transition is written in rock formations around the world. Red beds, sedimentary rocks stained by iron oxides, begin appearing in the geological record around 2.4 billion years ago. These formations represent the first time in Earth's history that iron could be oxidized and deposited on land surfaces exposed to the atmosphere. The red soils and rocks that formed during this period created some of the most visually striking evidence for the Great Oxidation Event. The banded iron formations that had characterized earlier periods began to disappear from the geological record as dissolved iron became scarce in oxygenated seawater. The last major banded iron formations date to around 1.8 billion years ago, marking the end of an era when iron-rich chemistry dominated marine environments. The oceans were transitioning from their ancient iron-rich state to something more recognizable as modern seawater. But the most dramatic evidence for rising oxygen levels comes from sulfur isotope signatures preserved in ancient rocks. Sulfur compounds show distinctive isotopic patterns that reflect whether they formed in oxygen-rich or oxygen-poor conditions. The isotopic signatures in rocks older than 2.4 billion years are completely different from those in younger rocks, providing clear chemical evidence for a fundamental change in atmospheric composition. The biological communities that had initiated this transformation faced their greatest challenge yet. The oxygen levels that were accumulating in the atmosphere were far higher than anything most organisms had evolved to tolerate. Many species that had thrived in the low oxygen conditions of earlier periods found themselves facing extinction as their environments became increasingly hostile to their anaerobic metabolism. The mass extinction that accompanied the Great Oxidation Event was one of the most severe in Earth's history. Entire lineages of anaerobic organisms disappeared from the fossil record, never to return. The stromatolite communities that had dominated shallow marine environments for over 1 billion years began to decline as oxygen-sensitive species were eliminated and oxygen-tolerant competitors gained advantages. Yet this crisis also created unprecedented opportunities for evolutionary innovation. The organisms that survived the oxygen transition found themselves in a world with vastly more chemical energy available for biological processes. Aerobic respiration could extract nearly 20 times more energy from organic compounds compared to anaerobic fermentation. This energy bonanza supported the evolution of larger, more complex cellular structures and more sophisticated metabolic capabilities. The development of aerobic respiration represented one of the most important biochemical innovations in the history of life. The process requires a complex chain of protein molecules that can safely transfer electrons from organic compounds to oxygen while capturing the released energy in forms that cells can use for growth and reproduction. The evolution of these electron transport systems required precise molecular engineering to prevent the formation of toxic oxygen radicals that could damage cellular components. The organisms that successfully mastered aerobic respiration gained enormous competitive advantages over their anaerobic relatives. They could grow faster, reproduce more frequently, and survive in environments where energy availability had previously limited biological activity. These aerobic organisms began to dominate ecosystems wherever oxygen was available, fundamentally altering the structure and function of biological communities. The cellular innovations that supported aerobic metabolism extended far beyond simple energy production. Cells had to evolve sophisticated systems for managing the toxic byproducts of oxygen metabolism, including highly reactive oxygen species that could damage proteins, lipids, and genetic material. The antioxidant systems that evolved during this period established the foundation for cellular protection mechanisms that remain essential in modern organisms. The compartmentalization of cellular processes became increasingly important as organisms developed ways to isolate oxygen-sensitive reactions from oxygen-rich parts of the cell. Some cells evolved complex internal membrane systems that created specialized environments for different metabolic pathways. These architectural innovations presage the elaborate cellular organization that would later characterize eukaryotic cells. The genetic regulatory systems that controlled these complex metabolic processes were becoming increasingly sophisticated. Cells needed precise mechanisms for sensing oxygen availability and adjusting their metabolic activities accordingly. The regulatory networks that evolved during this period could rapidly switch between aerobic and anaerobic metabolism depending on environmental conditions, providing crucial flexibility for surviving in a chemically dynamic world. The horizontal gene transfer that had been crucial for earlier evolutionary innovations continued to play important roles during the oxygen transition. Genes encoding oxygen tolerance mechanisms, aerobic respiratory systems, and antioxidant defenses spread rapidly through microbial populations, allowing beneficial adaptations to be shared across species boundaries. This genetic cooperation accelerated the pace of adaptation to oxygen-rich conditions. The ecological relationships within these evolving communities were becoming more complex as organisms developed new strategies for exploiting the energy-rich conditions created by atmospheric oxygen. Some species specialized in producing oxygen through photosynthesis, while others focused on consuming oxygen through aerobic respiration. These complementary metabolic strategies created opportunities for mutualistic relationships that benefited both partners. The predatory interactions that had begun during earlier periods intensified as aerobic metabolism provided the energy needed for more active hunting strategies. Some organisms evolved sophisticated mechanisms for detecting, pursuing, and capturing prey, while others developed defensive systems for avoiding predation. These biological arms races drove rapid evolution in both offensive and defensive capabilities. The symbiotic relationships that developed during this period would prove crucial for later evolutionary innovations. Some aerobic bacteria began living inside other cells, providing aerobic respiratory services in exchange for nutrients and protection. These endosymbiotic relationships established the foundation for the evolution of eukaryotic cells, which would revolutionize biological complexity in the next chapter of Earth's evolutionary story. The biogeochemical cycles operating during this period were fundamentally different from anything that had existed before. The carbon cycle was being driven by biological processes that could rapidly convert atmospheric carbon dioxide into organic compounds through photosynthesis, then release it back to the atmosphere through aerobic respiration and decomposition. This biological control of carbon cycling began to influence global climate patterns. The nitrogen cycle was also being transformed as organisms developed new pathways for processing nitrogen compounds in oxygen-rich environments. Nitrification processes that converted ammonia to nitrate became important sources of chemical energy for specialized bacteria, while denitrification processes that converted nitrate back to nitrogen gas helped regulate nitrogen availability in ecosystems. The phosphorus cycle remained largely controlled by geological processes, but biological organisms were developing increasingly efficient mechanisms for capturing and concentrating phosphorus from their environments. The competition for this essential but scarce element drove the evolution of sophisticated cellular machinery for phosphorus acquisition and storage. The iron cycle was being completely reorganized as dissolved iron became scarce in oxygenated waters. Organisms that had depended on iron-based metabolism were forced to develop alternative strategies or retreat to anoxic environments where iron remained available. The loss of bioavailable iron created new types of nutrient limitation that influenced ecosystem productivity and community structure. The trace element cycles that supported these complex biological processes were becoming more intricate as organisms developed requirements for specific metals and minerals that were essential for oxygen metabolism. Copper became crucial for enzymes involved in aerobic respiration, while manganese played important roles in protecting cells from oxidative damage. The biological demand for these trace elements began to influence their global cycling patterns. The seasonal patterns that characterized these oxygenated ecosystems reflected the complex interactions between biological oxygen production and consumption. Daily cycles of photosynthesis and respiration created fluctuations in local oxygen concentrations that organisms had to adapt to survive. Some species evolved circadian rhythms that synchronized their metabolic activities with these predictable environmental changes. The geographic distribution of oxygenated environments was initially patchy and heterogeneous, creating a complex mosaic of habitats with different chemical conditions. Organisms that could tolerate or utilize oxygen gained access to new territories, while those that required anoxic conditions became increasingly restricted to specialized refugia. This geographic reorganization of biological communities accelerated evolutionary divergence and speciation. The deep ocean basins remained largely anoxic for hundreds of millions of years after the atmosphere became oxygenated, providing refugia where ancient anaerobic lineages could persist and continue evolving. These oxygen-free environments became evolutionary museums that preserved metabolic pathways and cellular organization patterns that dated back to the earliest stages of life on Earth. The continental environments were experiencing oxygenation for the first time in Earth's history, creating entirely new types of habitats that had never supported life before. The weathering of rocks in oxygen-rich atmospheres produced new types of soils and sediments that could support biological communities. These terrestrial environments would eventually become sites of major evolutionary innovations, but during the immediate aftermath of the Great Oxidation Event, they remained largely uninhabited. The atmospheric chemistry that emerged from this biological revolution was unlike anything that had existed in the solar system before Earth's oxygen-rich atmosphere was a direct product of biological activity, representing the first time that living organisms had fundamentally altered the chemical composition of a planetary atmosphere. This biological signature would eventually become one of the most important indicators that scientists use to search for life on other worlds. The climate effects of atmospheric oxygenation were complex and sometimes contradictory. Oxygen itself is not a greenhouse gas, so its accumulation didn't directly warm the planet. However, the biological processes that produced oxygen also removed carbon dioxide from the atmosphere, potentially causing global cooling. The interactions between these different atmospheric changes created climate dynamics that were unlike anything Earth had experienced before. The ozone layer that formed when atmospheric oxygen was broken down by solar radiation provided crucial protection from harmful ultraviolet light that had previously sterilized Earth's surface environments. This UV shield made it possible for organisms to survive in shallow waters and eventually colonize terrestrial environments that had been too dangerous for life during earlier periods. The chemical weathering processes that operated in oxygen-rich atmospheres were fundamentally different from those that had characterized the anoxic world. The oxidation of minerals in rocks and soils released different types of nutrients and created different types of chemical environments. These changes in weathering patterns influenced the chemistry of rivers, lakes, and coastal marine environments. The preservation potential of organic matter was dramatically altered by the presence of atmospheric oxygen. Aerobic decomposition processes could completely break down organic compounds that would have been preserved under anoxic conditions, reducing the amount of organic carbon that was buried in sediments. This change in preservation efficiency influenced the global carbon cycle and the types of fossils that were preserved for future study. The evolutionary innovations that emerged during the Great Oxidation Event established many of the biochemical and ecological foundations that continue to support life on Earth today. The electron transport chains that power aerobic respiration, the antioxidant systems that protect cells from oxidative damage, and the regulatory mechanisms that control oxygen metabolism all trace their origins to adaptations that evolved during this crucial transition. The ecological principles that govern these early oxygenated ecosystems established patterns of energy flow and nutrient cycling that remain fundamental to modern ecosystem functioning. The relationships between primary producers, consumers, and decomposers that developed during this period created the basic architecture of food webs that continues to organize biological communities today. The biogeochemical cycles that emerged from this biological revolution continue to regulate Earth's environmental systems billions of years later. The oxygen cycle that maintains atmospheric oxygen levels, the carbon cycle that influences global climate, and the nitrogen cycle that supports biological productivity all depend on metabolic processes that evolved during the transition to an oxygenated world. The Great Oxidation Event represents one of the most profound examples of how biological processes can reshape planetary environments. The cyanobacteria that initiated this transformation didn't just adapt to their environment, they fundamentally altered the chemical and physical conditions on Earth in ways that created entirely new evolutionary possibilities. This demonstrates the incredible power of life to influence planetary evolution and highlights the intimate connections between biological and geological processes. The legacy of this ancient revolution continues to influence every aspect of life on Earth. The oxygen we breathe, the energy that powers our cells, and the protective ozone layer that shields us from harmful radiation all exist because of evolutionary innovations that emerged during this crucial transition period. The microscopic organisms that survived the Great Oxidation Event didn't just change their own world, they created the conditions that would eventually make complex multicellular life, including our own species, possible. The story of the Great Oxidation Event also provides important insights into the potential for biological processes to influence planetary environments on other worlds. As scientists search for signs of life beyond Earth, they look for atmospheric signatures similar to those created by the oxygen revolution. Recognizing that biological activity on the scale achieved by early cyanobacteria could create detectable changes in planetary atmospheric composition. The evolutionary creativity that emerged from the challenges of the oxygen transition demonstrates the remarkable adaptability of life in the face of environmental crisis. When faced with conditions that were toxic to most existing organisms, life didn't simply succumb to extinction. Instead, it evolved entirely new metabolic strategies that transformed environmental toxins into sources of abundant energy. This pattern of evolutionary innovation, in response to environmental challenge, has been repeated throughout the history of life on Earth. The Great Oxidation Event stands as one of the most important transitions in the history of our planet, marking the end of Earth's oxygen-free youth and the beginning of the oxygenated world that would eventually support the incredible diversity of complex organisms we see today. The microbial communities that initiated and survived this transformation had fundamentally altered the trajectory of evolution on Earth, creating possibilities that would unfold over the next 2 billion years of biological innovation and environmental change. Environmental change. The transformation that cyanobacteria had unleashed was now operating on a scale that dwarfed anything biology had achieved before. But this wasn't just about oxygen anymore. The success of these oxygen-producing communities had triggered a cascade of innovations that would reshape the very nature of life itself. The energy abundance created by aerobic metabolism was about to fund evolutionary experiments that seemed almost impossible under the harsh constraints of early Earth. The first hint of what was coming appeared in the cellular architecture of organisms that had mastered the art of oxygen tolerance. Some of these cells were growing larger than anything that had existed before, developing internal structures that looked suspiciously organized, instead of the simple, undifferentiated interiors that had characterized prokaryotic cells for billions of years. These organisms were experimenting with compartmentalization on an unprecedented scale. The driving force behind this cellular revolution was the same energy crisis that had powered earlier innovations. Larger cells faced a fundamental problem. As cellular volume increased, the surface area available for exchanging materials with the environment increased much more slowly. This created a bottleneck that limited how large and complex cells could become while still maintaining the chemical gradients needed for life. The solution that emerged was nothing short of revolutionary. some cells began developing elaborate internal membrane systems that dramatically increased their internal surface area. These weren't just random folds in the cell wall. They were organized structures that created specialized internal environments for different cellular processes. The most successful of these experiments involved membranes that could isolate oxygen-sensitive reactions from oxygen-rich parts of the cell while maintaining efficient transport of materials between different compartments. But the real breakthrough came when some of these large complex cells began incorporating smaller cells as permanent residents. This wasn't predation or parasitism in the traditional sense. These were cooperative relationships where different organisms contributed specialized services to a shared cellular community. The smaller residents provided metabolic capabilities that the host cell couldn't perform on its own, while the host provided protection and resources that the residents couldn't access independently. The most successful of these cellular partnerships involved aerobic bacteria that had evolved highly efficient oxygen-consuming metabolism. When these organisms took up residence inside larger cells, they created biological power plants that could generate enormous amounts of energy from organic compounds and oxygen. The host cells could tap into this energy source, while the bacterial residents enjoyed a stable, resource-rich environment where they were protected from environmental fluctuations and threats. These endosymbiotic relationships required extraordinary levels of biochemical coordination. The bacterial residents had to synchronize their reproduction with that of their hosts to maintain optimal population sizes. They had to develop transport systems for sharing metabolic products with their hosts while maintaining their own cellular integrity. Most remarkably, they had to evolve mechanisms for transferring some of their genetic material to their hosts, creating integrated genetic systems that could coordinate the activities of what had once been independent organisms. The evolutionary implications of this cellular merger were staggering. For the first time in Earth's history, individual organisms could combine the metabolic capabilities of multiple different species within a single cell. This opened up entirely new ecological niches and metabolic strategies that had been impossible when organisms were limited to their own biochemical capabilities. The energy budget available to these composite cells was orders of magnitude higher than what any single organism could achieve independently. The success of endosymbiotic cells created positive feedback loops that accelerated further innovation. The abundant energy provided by their internal power plants allowed these organisms to invest in larger and more complex cellular structures. They could afford to maintain elaborate genetic systems, sophisticated protein synthesis machinery, and complex regulatory networks that would have been energetically prohibitive for simpler cells. The genetic innovations that emerged from these cellular partnerships were equally revolutionary. The transfer of genetic material between endosymbionts and their hosts created opportunities for genetic recombination that had never existed before. Instead of relying solely on horizontal gene transfer between independent organisms, these composite cells could shuffle genetic material internally, creating new combinations of genes and regulatory systems within individual lineages. The development of sexual reproduction represented the ultimate expression of this genetic innovation. Some of these complex cells evolved the ability to combine genetic material from two different individuals, creating offspring that carried genetic contributions from multiple parents. This process dramatically accelerated the pace of evolutionary innovation by creating new genetic combinations in every generation, rather than relying on random mutations as the sole source of genetic variation. The cellular architecture that supported sexual reproduction required sophisticated molecular machinery for managing genetic material. These cells had to evolve mechanisms for packaging their genetic material into discrete units that could be precisely divided and distributed during cell division. They needed quality control systems that could detect and repair errors in genetic copying. Most remarkably, they had to develop timing mechanisms that could coordinate the complex sequence of events involved in genetic recombination and cell division. The emergence of these complex cells marked the beginning of the eukaryotic revolution that would eventually lead to all complex multicellular life on Earth. but the immediate impact was more subtle. These early eukaryotic cells were still single-celled organisms living in microbial communities that looked superficially similar to the prokaryotic ecosystems that had dominated Earth for billions of years. The real changes were happening at the molecular level, where new types of cellular organization were being tested and refined. The metabolic capabilities of these early eukaryotic cells were becoming increasingly sophisticated as they learned to exploit the energy abundance provided by their endosymbiotic partners. Some species developed the ability to switch between different metabolic strategies depending on environmental conditions, using aerobic respiration when oxygen was abundant, but reverting to fermentation when oxygen levels dropped. This metabolic flexibility provided crucial advantages in environments where oxygen availability fluctuated unpredictably. The cellular division mechanisms employed by these complex cells were far more elaborate than the simple binary fission used by prokaryotic organisms. The process of dividing genetic material between daughter cells required precise molecular machinery that could ensure each new cell received a complete set of genetic instructions. The evolution of this cellular division system established the foundation for the precise genetic inheritance that would become essential for complex multicellular development. The size advantages that eukaryotic cells could achieve opened up entirely new ecological possibilities. Larger cells could engulf and consume smaller organisms more effectively than prokaryotic cells, leading to the evolution of more sophisticated predatory strategies. Some eukaryotic cells became specialized hunters that actively pursued and captured prey, while others developed defensive mechanisms for avoiding predation by larger organisms. The internal transport systems that evolved within these complex cells were marvels of cellular engineering. These organisms had to develop molecular highways that could move materials efficiently between different cellular compartments while maintaining the chemical gradients needed for proper cellular function. The evolution of these transport systems required sophisticated molecular motors and targeting mechanisms that could deliver specific molecules to precise cellular locations. The protein synthesis machinery that operated within eukaryotic cells was becoming increasingly complex as these organisms evolved larger and more diverse protein repertoires. The cellular structures responsible for making proteins had to coordinate with genetic control systems that could regulate when and where different proteins were produced. This coordination required elaborate communication networks that could integrate information about cellular needs with genetic instructions for protein synthesis. The waste management systems operating within these large cells had to handle the increased metabolic byproducts generated by more active cellular metabolism. These organisms evolved sophisticated mechanisms for detoxifying harmful compounds, recycling cellular components, and disposing of materials that could interfere with cellular processes. The evolution of these cellular housekeeping systems was essential for maintaining the internal organization that made complex cellular architecture possible. The environmental sensing systems that guided eukaryotic cell behavior were becoming more sophisticated as these organisms developed the ability to detect and respond to increasingly subtle environmental cues. Some cells evolved sensory mechanisms that could detect chemical gradients, light levels, or physical forces in their environment. These sensory capabilities allowed eukaryotic cells to actively navigate their environment and seek out favorable conditions. The cellular communication systems that operated between different eukaryotic cells were evolving greater complexity as these organisms began experimenting with cooperative behaviors that required coordination between multiple individuals. Some species developed the ability to produce chemical signals that could trigger coordinated responses in nearby cells, allowing groups of individual cells to behave as integrated communities. The defensive systems that protected eukaryotic cells from environmental threats were becoming more elaborate as these organisms faced increasing challenges from predators, parasites, and environmental toxins. Some cells evolved the ability to produce protective compounds that could deter predators or neutralize toxins. Others developed rapid response mechanisms that could quickly isolate and contain threats before they could cause cellular damage. The reproductive strategies employed by eukaryotic cells were diversifying as these organisms experimented with different approaches to genetic inheritance and population maintenance. Some species maintained the simple, asexual reproduction strategies used by prokaryotic organisms, while others evolved complex sexual reproduction systems that involved genetic recombination between multiple individuals. The dispersal mechanisms used by eukaryotic cells were becoming more sophisticated as these organisms developed better strategies for colonizing new environments and maintaining genetic connectivity between distant populations. Some species evolved resistant spores that could survive harsh environmental conditions during transport, while others developed active swimming capabilities that allowed them to actively migrate to new habitats. The symbiotic relationships that eukaryotic cells formed with other organisms were becoming increasingly diverse and specialized as these complex cells explored new forms of biological cooperation. Some partnerships involved metabolic cooperation, where different organisms contributed complementary biochemical capabilities. Others involved protective relationships, where different species provided mutual defense against environmental threats. The competitive interactions between eukaryotic cells and prokaryotic organisms were intensifying as these two fundamentally different types of cellular organization competed for resources and ecological niches. The larger size and higher energy budgets of eukaryotic cells provided advantages in some environments, while the metabolic efficiency and rapid reproduction of prokaryotic cells remained advantageous in others. The ecosystem engineering activities of eukaryotic cells were beginning to influence their local environments in new ways as these larger, more active organisms modified their surroundings through their metabolic activities and behaviors. Some eukaryotic cells developed the ability to create structures that modified local water flow patterns or chemical gradients in ways that favored their own survival and reproduction. The biogeochemical impacts of early eukaryotic cells were still relatively modest compared to the massive environmental changes that had accompanied earlier evolutionary innovations. However, the foundation was being laid for future developments that would eventually allow eukaryotic organisms to become dominant forces in global environmental systems. The preservation potential of early eukaryotic cells in the fossil record was limited by their larger size and more complex cellular organization, which made them more susceptible to decay and less likely to be preserved as recognizable fossils. However, some exceptional preservation sites have yielded remarkable fossils that provide direct evidence for the cellular complexity that had evolved by this time. The molecular fossils that represent early eukaryotic cells include distinctive organic compounds that are produced only by organisms with complex cellular organization. These biomarker molecules can survive in ancient rocks long after cellular structures have been destroyed, providing chemical evidence for the presence of eukaryotic cells in ancient environments. The isotopic signatures associated with early eukaryotic metabolism show distinctive patterns that reflect the more complex biochemical processes operating within these cells. The carbon isotope ratios produced by eukaryotic cells are often different from those produced by prokaryotic organisms, allowing scientists to track the emergence and expansion of eukaryotic life in the geological record. The trace fossil evidence for early eukaryotic behavior includes structures that reflect the larger size and more active lifestyle of these organisms compared to prokaryotic cells. Some ancient sediments contain traces that appear to have been made by organisms large enough and active enough to be eukaryotic cells, providing indirect evidence for their presence and activities. The phylogenetic relationships between early eukaryotic cells and modern organisms can be reconstructed using comparative analysis of genetic sequences from living species. These molecular evolutionary studies reveal that the fundamental cellular organization that characterizes all eukaryotic cells evolved during this ancient period, and has been conserved throughout subsequent evolution. The experimental studies that recreate the environmental conditions present during early eukaryotic evolution provide insights into the chemical and physical processes that could have supported the emergence of complex cellular organization. These laboratory experiments help scientists understand how endosymbiotic relationships could have evolved and how the metabolic integration between different cellular partners could have been achieved. The comparative studies of modern organisms that retain primitive characteristics similar to early eukaryotic cells provide valuable insights into how these ancient organisms might have functioned. Some modern protists show cellular organization patterns that may resemble those of early eukaryotic cells, serving as living models for understanding ancient biology. The environmental conditions that supported early eukaryotic evolution were characterized by increasing oxygen availability and more stable chemical conditions compared to earlier periods in Earth's history. The development of more predictable environmental conditions provided the stable foundation needed for the evolution of complex cellular organization that required precise coordination between multiple cellular components. The geographic distribution of early eukaryotic cells was initially limited to environments where oxygen levels were sufficient to support aerobic metabolism, but not so high as to create dangerous levels of oxidative stress. These organisms gradually expanded their geographic range as they evolved better mechanisms for tolerating and utilizing oxygen-rich conditions. The temporal patterns of early eukaryotic evolution reflect the gradual nature of the cellular innovations that led to complex cellular organization. The fossil record suggests that the transition from prokaryotic to eukaryotic cellular organization occurred over hundreds of millions of years, with different aspects of cellular complexity evolving at different rates and in different lineages. The evolutionary constraints that influenced early eukaryotic evolution included the need to maintain compatibility between different cellular components, while simultaneously evolving greater complexity and efficiency. The organisms that successfully navigated these constraints established the cellular architecture that would provide the foundation for all subsequent eukaryotic evolution. The adaptive radiation that followed the establishment of eukaryotic cellular organization created the first major diversification of complex cells, with different lineages exploring different ecological niches and metabolic strategies. This early diversification established many of the major groups of eukaryotic organisms that continue to exist today. The coevolutionary relationships between eukaryotic cells and their prokaryotic endosymbionts drove continued innovation in cellular organization and metabolic capabilities. The ongoing evolutionary refinement of these cellular partnerships led to increasingly efficient and sophisticated cellular systems that could exploit environmental resources more effectively than either partner could achieve independently. The metabolic innovations that emerged from eukaryotic cellular organization established new biogeochemical pathways that would eventually influence global environmental systems. Although the immediate impact of early eukaryotic cells on global chemistry was modest, they were establishing the metabolic foundations that would later support much larger and more influential biological communities. The cellular complexity that characterized early eukaryotic cells represented a fundamental transition in the organization of life on Earth. For the first time, individual cells contained multiple different types of cellular machinery that could work together to achieve capabilities that exceeded what any single cellular system could accomplish. This principle of biological integration through cooperation would later become essential for the evolution of multicellular organisms. The energy budgets available to eukaryotic cells were orders of magnitude higher than those available to prokaryotic cells, providing the metabolic foundation needed for the evolution of larger, more complex biological structures. This energy abundance would eventually support the evolution of multicellular organisms with specialized tissues and organs. But those developments were still hundreds of millions of years in the future. The genetic systems that evolved within eukaryotic cells established new mechanisms for storing, processing, and transmitting biological information. The combination of larger genomes, more sophisticated genetic regulation, and sexual reproduction created unprecedented opportunities for evolutionary innovation and adaptation to changing environmental conditions. The cellular architecture that emerged from early eukaryotic evolution established the basic organizational principles that continue to characterize all complex cells today. The compartmentalization of cellular functions, the integration of endosymbiotic organelles, and the sophisticated molecular machinery that coordinates cellular activities all trace their origins to innovations that evolved during this crucial period. The evolutionary success of eukaryotic cells demonstrates the power of cooperation and integration in biological systems. The organisms that achieved the greatest success during this period were those that could most effectively coordinate the activities of multiple different cellular components, while maintaining the flexibility needed to adapt to changing environmental conditions. The transition to eukaryotic cellular organization represents one of the major evolutionary transitions in the history of life on Earth, comparable in importance to the origin of life itself and the evolution of multicellular organisms. This transition established the cellular foundation that would eventually support all complex life forms, from microscopic protists to the largest plants and animals. The legacy of early eukaryotic evolution continues to influence every aspect of biology today. The cellular organization patterns that evolved during this ancient period provide the structural foundation for all complex organisms, while the metabolic pathways that emerged from endosymbiotic relationships continue to power the majority of biological processes on Earth. The story of early eukaryotic evolution illustrates the creative potential of evolutionary processes when environmental conditions provide both challenges and opportunities for biological innovation. The organisms that emerged from this evolutionary transition didn't just adapt to their environment. They developed entirely new ways of being alive that opened up evolutionary possibilities that had never existed before. The cellular complexity that characterized these early eukaryotic organisms was setting the stage for even greater innovations that would unfold over the next billion years of evolution. The metabolic capabilities, genetic systems, and cellular architecture that had evolved during this period would provide the foundation for the evolution of multicellular organisms, complex developmental processes, and eventually the incredible diversity of complex life forms that characterize modern ecosystems. But those developments were still far in the future. For now, in the gradually oxygenating oceans of the Proterozoic world, these remarkable cellular innovations were being tested and refined by natural selection. The eukaryotic cells that had emerged from the endosymbiotic revolution were establishing new patterns of biological organization that would prove crucial for the next great chapter in the evolution of life on Earth. The stage was set for complexity on a scale that the ancient world had never imagined. The microscopic world that emerged from these cellular innovations was quietly revolutionizing the very meaning of biological complexity. These early eukaryotic organisms weren't just larger or more sophisticated than their prokaryotic ancestors. They represented an entirely new approach to being alive, one that would eventually make possible every complex organism that has ever existed on Earth. The cellular partnerships that had given birth to eukaryotic life were becoming increasingly refined through millions of generations of evolutionary fine-tuning. The bacterial endosymbionts that had initially been independent organisms were gradually losing their autonomy, transferring more and more of their genetic material to their host cells while becoming increasingly specialized for their internal roles. This genetic integration created cellular systems that were far more than the sum of their parts. The mitochondria that evolved from these ancient bacterial partnerships became the powerhouses of eukaryotic cells, but their transformation was gradual and complex. Over time, these organelles shed most of their original genes, retaining only those absolutely essential for their energy-producing functions. The genes they lost weren't destroyed. They were transferred to the nucleus of their host cells, creating integrated genetic systems that required precise coordination between nuclear and organella gene expression. This genetic integration had profound implications for how eukaryotic cells could evolve and adapt to changing conditions. Unlike prokaryotic cells that relied primarily on horizontal gene transfer for acquiring new capabilities, eukaryotic cells could evolve through the gradual modification of their integrated genetic systems. This created opportunities for more sophisticated evolutionary fine-tuning, but it also made eukaryotic evolution more conservative and less rapid than prokaryotic evolution. The energy production systems that operated within these early eukaryotic cells were becoming increasingly efficient as the coordination between nuclear and mitochondrial functions improved. The cellular machinery responsible for aerobic respiration was being refined through countless generations of selection, creating biochemical systems that could extract maximum energy from available resources while minimizing the production of harmful waste products. The cellular transport systems that moved materials between different compartments within eukaryotic cells were evolving remarkable sophistication. These weren't just simple diffusion processes, but active transport networks that could selectively move specific molecules to precise cellular locations. The evolution of these transport systems required the development of molecular motors, targeting signals, and quality control mechanisms that ensured cellular components ended up in the right places at the right times. The protein synthesis machinery operating within eukaryotic cells was becoming far more complex than anything that had existed in prokaryotic organisms. The ribosomes responsible for making proteins were larger and more sophisticated, capable of producing more complex proteins with greater precision. The cellular systems responsible for modifying and processing these proteins after they were made were equally elaborate, creating cellular factories that could produce a remarkable diversity of molecular tools. The genetic regulatory systems that controlled eukaryotic cellular functions were evolving unprecedented complexity. Unlike prokaryotic cells that primarily used simple on-off switches to control gene expression, eukaryotic cells were developing regulatory networks that could integrate multiple different types of information to make sophisticated decisions about cellular activities. These systems could respond to internal cellular conditions, external environmental cues, and developmental programs that coordinated cellular activities over long time periods. The development of sexual reproduction in some eukaryotic lineages represented one of the most significant innovations in the history of life. This process allowed organisms to combine genetic material from two different individuals, creating offspring that carried new combinations of genes in every generation. The molecular machinery required for sexual reproduction was extraordinarily complex, involving precise mechanisms for genetic recombination, chromosome segregation, and gamete formation. The advantages provided by sexual reproduction were not immediately obvious, since the process required significant energy investment and reduced the reproductive output of individual organisms. However, sexual reproduction dramatically accelerated the pace of evolutionary adaptation by creating genetic variation much faster than mutation alone could achieve. This innovation would prove crucial for allowing eukaryotic organisms to adapt rapidly to changing environmental conditions. The cellular division processes used by eukaryotic cells were far more elaborate than the simple binary fission employed by prokaryotic organisms. The process of mitosis required sophisticated molecular machinery that could precisely duplicate and distribute genetic material between daughter cells. This system had to coordinate the replication of nuclear DNA, the division of organelles, and the physical separation of cellular contents in a carefully orchestrated sequence of events. The size advantages that eukaryotic cells achieved through their complex internal organization opened up entirely new ecological niches. These larger cells could engulf and digest smaller organisms more effectively than prokaryotic cells, leading to the evolution of sophisticated predatory strategies. Some early eukaryotes became specialized hunters that actively pursued prey, while others developed filter feeding mechanisms that could capture large quantities of small organisms. The defensive capabilities of eukaryotic cells were evolving in response to increasing predation pressure from other eukaryotic organisms. Some cells developed protective shells or cases that made them difficult to digest, while others evolved toxic compounds that made them unpalatable to predators. The co-evolutionary arms race between predators and prey was driving rapid innovation in both offensive and defensive cellular capabilities. The motility systems that allowed eukaryotic cells to move through their environment were becoming increasingly sophisticated. Some organisms developed flagella or cilia that could propel them through water with remarkable efficiency, while others evolved amoeba-like movement systems that allowed them to crawl across surfaces. These locomotory capabilities provided significant advantages for finding food, avoiding threats, and locating suitable reproductive partners. Tonight, we've travelled back through 3.5 billion years to witness something extraordinary. In the most hostile conditions imaginable, life didn't just survive, it thrived and transformed our entire planet. From those first chemical experiments around hydrothermal vents to the revolutionary communities that learned to harness sunlight, these ancient pioneers laid the foundation for everything that followed. Every breath you take connects you to those microscopic heroes who turned poison into power and chaos into complexity. The story of life's beginning reminds us that even in the darkest moments, innovation finds a way.